So here is something I would like you to do right now as you settle in for the evening. Think of a smell, any smell from your childhood. Maybe it is your grandmother's kitchen. Maybe it is wet pavement after a summer rain, or a Christmas tree. The smell of tangerines. A smell you have not encountered in years, maybe decades. But right now, just hearing those words, something stirred inside you. You may not have simply remembered the smell. You remembered the light in the room, someone's face, maybe the texture of the tablecloth, or the feeling of a cold floor under bare feet. And chances are, along with all of that, an emotion arrived. Warm, soft, maybe a little bit sad. You did not choose that emotion. It simply appeared alongside everything else, as though someone pressed a button and launched an entire world. All of that took less than a second, maybe half a second, maybe even less. But in that tiny fraction of time, something staggeringly complex happened inside your head. Thousands of neurons in your visual cortex reproduced a fragment of an image you saw decades ago. Your auditory cortex quietly activated the ghost of a sound. Your olfactory cortex conjured a hint of a smell that is not actually there. The hippocampus, a structure about the size of your little finger, tucked deep inside your brain, pulled all of those fragments together, like a dispatcher who remembers which shelves hold the pieces of a puzzle that was taken apart long ago. The amygdala added the emotional coloring, and that is exactly why the memory arrived with a feeling rather than as a dry list of facts. And at the deepest level, at a level you cannot see under any ordinary microscope, the configuration of proteins around your DNA shifted. Chemical tags on the chromatin of your neurons moved. Genes that had been closed cracked open. RNA molecules set off on a journey from the nucleus of the cell to the synapse, the point of contact between neurons, carrying instructions that will slightly alter that contact. And here is what is remarkable. By the time that journey is complete, the memory you just recalled will be recorded slightly differently than it was before this moment. You literally rewrote a small fragment of yourself while lying here thinking about your grandmother's kitchen. Tonight, we are going to take that single second apart. We are going to do it slowly, layer by layer, from what you feel when you remember, all the way down to the molecules that make it possible. We will walk through every floor of this structure, from the cortex, where the image is restored, through the hippocampus, which knows where to find its fragments, through the synapses, where the connections between neurons are stored, all the way down to the deepest level, to the DNA, to the chemical tags on your genes that hold the record of your experience in a code that evolution has been refining for 4 billion years. And along the way, we will answer a few questions that I think are worth asking. How does your brain decide which moments become memories and which ones vanish forever? Why do you still remember the smell of tangerines from your childhood, but cannot recall what you had for lunch on Tuesday? Where exactly does a specific memory live, and can you actually reach it, touch it, switch it on or off? What happens to the molecules in your brain when you remember something? Why does the same memory feel different every time you return to it? And if your memories are constantly being rewritten every time you summon them, what does that mean about who you really are? There is no rush with any of this. We have plenty of time, you and I. We will move slowly, as if we are taking apart a nesting doll, removing one shell after another, and each one inside will be smaller and more surprising than the last. And if you happen to drift off somewhere along the way, that is perfectly fine. The story will still be here when you come back. Let us start with the outermost layer, with what a memory feels like when it arrives. Marcel Proust opened one of the most famous novels of the 20th century with a scene in which the protagonist takes a bite of a small sponge cake, a madeleine, dipped in a cup of linden tea. And that taste triggers an avalanche. An entire world of childhood memories crashes over him. His aunt's house in Combray, the garden, the streets of the little town, the church, the people, the smells, the light. All of it had been buried somewhere inside him for decades, and a single sip of tea with cake was enough to bring it all rushing back. Proust described this with brilliant precision, though he had no idea, of course, what was actually happening inside his brain at that moment. He simply captured something every one of us knows. Memories do not sit neatly folded on shelves, waiting for us to come and collect them. They hide, sometimes for decades, and they return when you least expect them, summoned by a smell, a sound, a passing phrase, a melody you have not heard in 20 years. The physicist Brian Greene, host of the World Science Festival, once told the story of his earliest memory. He is in a stroller, his father pushing him down 81st Street in Manhattan, and he is playing with beads on a bar in front of him. His father pushes them to the corner of 82nd and Columbus, to his favorite hot dog stand. Greene remembers the warmth of the bun, the crunch of that first bite, the bumps of the sidewalk under the stroller wheels. But then he says something important. I see this scene in third person. I am watching myself from above. He sees a little boy in the stroller, but he does not see the world from inside the stroller. The camera has shifted. Somehow, over the decades, over all the times he told that story to his children and his friends, his brain re-edited the memory. The event was real. His father really did buy him hot dogs at the corner of 82nd and Columbus. But the film that his brain plays for Greene now is not the recording that the camera made that day. It is a reconstruction. Reassembled many times over, supplemented, slightly altered. And there is the first hint of what happens to a memory every time we call it up. But we will get to that. First, let us sort out what kinds of memories there actually are. Because the word memory is deceptively uniform. It sounds as though we are talking about a single phenomenon. You might say, I have a good memory, or my memory is getting worse. But when you say the word memory, you are actually talking about an entire family of completely different processes, living in different parts of the brain, operating by different rules, and even breaking down in different ways. Imagine you are riding a bicycle. You are not thinking about how to pedal, how to keep your balance, how to turn the handlebars. It all happens automatically. Once, as a child, you learned how, and it was hard. You fell, your parents ran alongside holding the seat, but then something clicked, and from that point on, you could simply ride a bicycle. You cannot even properly explain how you do it. If someone asked you to describe in words exactly how you maintain balance, you would start stumbling over your answer. Because that knowledge does not live in words.
It lives in your muscles, in your cerebellum, in your basal ganglia, structures that handle practiced movements. Now imagine a different kind of memory. Someone asks you, "What is photosynthesis?" And you answer, "It is the process by which plants use sunlight to convert carbon dioxide and water into glucose and oxygen." You know that fact. You can say it out loud. But you most likely do not remember when exactly you learned it. Maybe 7th grade? Maybe from a textbook? Maybe somebody told you? The fact detached from the specific episode in which you first encountered it and became part of your general knowledge about the world. Neuropsychologists call this semantic memory. Memory for facts, definitions, word meanings, general knowledge. It lives primarily in the anterior portions of the temporal lobes, and it is a different system from the one that stores specific episodes of your life. When you remember the actual day you first learned about photosynthesis, if you can remember it at all, that is episodic memory. Personal, autobiographical, anchored to a place and a time. You remember being in a classroom, that it was raining outside the window, that the teacher was saying something about chlorophyll, and you were bored, and you were doodling in your notebook. That memory, with its place, time, sensations, and emotions, lives in a different system, and that system depends critically on the hippocampus. There is also working memory, the thing that lets you hold a phone number in your head for a few seconds while you dial it. If something distracts you, the number vanishes. Working memory is not built for long storage. It is like a whiteboard where you write in marker and erase after a minute. Scientists associate it mainly with the prefrontal cortex, the part of the brain located right behind your forehead.
All of these systems run simultaneously, and you usually do not even notice switching between them. When you are driving a familiar route, procedural memory, while thinking about what to cook for dinner, working memory, listening to the radio and recognizing the name of a politician, semantic memory, and suddenly remembering you forgot to pick up a package from the post office yesterday, episodic memory. Your brain is running 4 different memory systems in parallel, each with its own anatomy, its own biochemistry, its own rules. And here is what is especially interesting. These systems can break down independently. A person can lose the ability to form new episodic memories while still learning new motor skills perfectly well. They can forget names and faces but remember how to play the piano. They can have no idea what year it is but still know that Paris is the capital of France.
It was precisely these cases of selective memory impairment that allowed scientists in the second half of the 20th century to figure out that memory is not a single function, but an entire family, and every member of that family lives at a different address. But before scientists understood this, they had to travel a long road. And on that road, there were crude mistakes. There were brilliant guesses. And there was one patient whose tragedy changed everything we know about how the brain creates memories. A patient who spent most of his life stranded in a permanent present, and who, without ever knowing it, gave humanity one of the most important clues about the nature of memory. His name was Henry Molaison, and his story begins with an operation that went wrong. To understand where memories live, humans needed centuries, and for most of those centuries, they were looking in the wrong place.
For the greater part of the history of Western medicine, the brain was not even considered the organ responsible for thinking and memory. Aristotle, in the 4th century BC, believed the brain was simply a cooling device for the blood, something like a radiator. The heart does the thinking, Aristotle argued. The brain just keeps the blood from overheating. And that idea held on for a surprisingly long time, partly because Aristotle was so authoritative that people did not argue with him for centuries. But there were others who understood from the very beginning that the brain was the command center. Hippocrates, 100 years older than Aristotle, wrote directly that the brain is the interpreter of consciousness. Galen, in the 2nd century AD, developed this idea further, connecting brain injuries to disturbances of sensation and movement. But Galen, like everyone before him, was limited by his tools. He could observe that a blow to the head causes loss of consciousness. He could see that damage to certain areas of the brain leads to paralysis in certain parts of the body. But how exactly the brain does what it does remained a mystery. The first truly systematic attempt to map the functions of the brain came from an unexpected and frankly dubious source. In the early 19th century, the German physician Franz Joseph Gall proposed a theory he called phrenology. Gall's idea was simple and, at first glance, appealing. Different mental functions, memory, language, musical ability, aggression, are located in different parts of the brain. And if a particular function is especially well developed, the corresponding area of the brain grows larger, and this enlargement is reflected in the shape of the skull. Therefore, by feeling the bumps and hollows on someone's head, you could determine their character and abilities. Today we know this is nonsense. The shape of the skull does not reflect the shape of the brain, and the brain does not work as a set of separate modules with sharply drawn boundaries. But Gall did get one thing right, and it is precisely for that one thing that he deserves his place in history. He was the first person to seriously ask whether different parts of the brain might be responsible for different functions. The idea of localization, despite the wrongness of Gall's particular implementation, turned out to be fruitful. Real evidence that specific functions are tied to specific brain regions began appearing in the middle of the 19th century, most often coming from clinical medicine, from doctors working with patients whose brains had been damaged by injury or disease. In 1861, Paul Broca presented to the Anthropological Society of Paris the case of a patient named Le Borgne, whom the hospital staff had nicknamed Tan, because the only thing he could say was that single syllable. Tan, tan.
Le Borgne understood speech. He could follow instructions, but he could not speak. When Le Borgne died and Broca examined his brain, he found damage in the posterior part of the left frontal lobe. That region has been called Broca's area ever since, and we know it is critically important for the production of speech. A few years later, Carl Wernicke described the opposite case. A patient who could speak fluently, uttering long, grammatically correct sentences, but whose speech was meaningless. He could not understand what was said to him and did not understand his own words. The damage was in the posterior part of the left temporal lobe, in the area now called Wernicke's area. One region for producing speech, another for understanding it. The brain truly does have a functional map. But mapping memory turned out to be considerably harder. The turning point came in the 1930s and 40s, and it involved a neurosurgeon named Wilder Penfield, who worked at the Montreal Neurological Institute. Penfield operated on patients with severe epilepsy, and to locate the precise source of their seizures, he stimulated different areas of the brain's cortex with weak electrical impulses during surgery. The patients were conscious throughout. The brain has no pain receptors, and brain surgery can be performed under local anesthesia while the patient is awake and talking with the surgeon. Penfield discovered something astonishing. When he stimulated certain areas of the temporal lobe, patients reported vivid, detailed memories. One woman suddenly heard a Christmas song she had not heard in many years. Another patient saw a scene from childhood, a specific evening, a specific room. These were not vague sensations. They were specific, alive, richly detailed experiences, triggered by the touch of an electrode to a particular point on the surface of the brain. Penfield was stunned. He concluded he had found the storage vault of memory, that memories were recorded on the surface of the cortex like grooves on a phonograph record, and his electrode was the needle dropping into the right groove. He was not entirely right, as we will see, but his experiments proved something fundamental. Stimulation of specific brain areas can produce the subjective experience of a memory. Memory really is stored in the brain, in physical substrate, and it can be accessed by physical means. But the true revolution in understanding how the brain handles memory came about because of a single surgical catastrophe. And at the center of that catastrophe stood a 27-year-old man from Connecticut, known in the scientific literature for decades only by the initials HM, to protect his privacy. His full name, Henry Gustav Molaison, became public only after his death in 2008. Henry had been suffering from severe epileptic seizures since the age of 10. By 27, the seizures had become so frequent and debilitating that he could not work, could not live a normal life. The medications of the time were no help. In 1953, the neurosurgeon William Beecher Scoville proposed a radical operation. Remove the medial temporal lobe structures from both sides of the brain, including most of both hippocampi. The logic was simple. If the source of the epilepsy lies in these structures, remove them and the seizures will stop. The operation did help with the epilepsy. The seizures became significantly less frequent and less severe. But the cost was unimaginable. When Henry came to after surgery, he could talk. He remembered his childhood. He remembered events from years before the operation. His IQ actually rose slightly, probably because the constant seizures were no longer interfering with his brain's functioning. His personality was unchanged. He was polite, friendly, cooperative. But he could not remember anything new. If you walked into his room, introduced yourself, talked with him for 10 minutes, stepped out, and came back half an hour later, he had no idea who you were. He did not remember you had visited. For him, every meeting was the first. Brenda Milner, a neuropsychologist from the Montreal Neurological Institute, worked with Henry for decades. She met him hundreds of times. He never once recognized her. Every visit from Brenda Milner was, for Henry, the first. Henry could not remember a newspaper he had just read. He could read the same article over and over, and each time it was new to him. He did not know his parents had died. Every time he was told, he grieved as though hearing it for the first time. He did not know what year it was. He did not know how old he was. In some deep sense, he was permanently locked in 1953, in the day before the surgery. The world around him changed, but he stayed in place. Think about that for a moment. Henry Molaison lived another 55 years after his operation. 55 years in which he could not add a single new episodic memory. 55 years without accumulating new experience, without building a new biography, without the continuous stream of events that makes our lives ours. But here is what made the case of HM invaluable to science. His memory loss was selective. He had not lost all memory. He had lost the ability to form new declarative memories, new episodes, new facts. But other kinds of memory remained untouched. Brenda Milner ran a famous experiment. She asked Henry to trace the outline of a star while looking only in a mirror. If you have ever tried this, you know it is incredibly difficult at first.
The mirror reflection reverses all your movements, and you draw lines in the wrong directions. But with practice, you learn. You get better. Your hand starts moving more confidently. The lines get straighter. Henry got better too. With each session, his star drawing was more accurate. His motor system was learning. His brain was storing the skill. But every time Milner brought him the mirror and the pencil, he did not remember ever having done this before. He was surprised that he was so good at mirror drawing on what he believed was his very first attempt. Of course, it was not the first attempt. His hands remembered. His consciousness did not. Let us stay with this, because the life of Henry Molaison after his surgery deserves a closer look. His case did not simply answer the question of where memory lives in the brain. It showed, with frightening clarity, what it means to live without the ability to create new memories.
And it showed something else. Something that forced neuroscientists to completely rethink the very nature of memory. Every morning, Henry woke up and did not know what day it was. He could read the morning newspaper and 10 minutes later read it again, with the same interest, the same reactions to the same headlines. He could talk with a nurse about the weather, leave the room, come back 5 minutes later, and start the same conversation over. For the nurse, it was a repeat. For Henry, it was the first time, every time. But Henry was not unhappy, at least he did not appear to be. He was unfailingly friendly, patient, cooperative. He knew something was wrong with him. He understood that his memory was impaired, but he could not grasp the depth of the impairment, because to grasp the depth of a loss, you need the ability to compare the present with the past, and his present never anchored itself in the past. Every moment was the only moment, and then it vanished.
Larry Squire, who described this case in detail in his 1987 book, Memory and Brain, highlighted several key observations that Brenda Milner and her colleagues made while working with Henry. First, Henry's old memories were largely intact. He remembered his childhood. He remembered where he grew up. He remembered events from his life before the operation, although memories from the years immediately preceding the surgery were somewhat blurred, a phenomenon neuropsychologists call temporally graded retrograde amnesia. The closer to the moment of surgery, the hazier the memories. The further back in time, the more reliable they were. This was an important detail. It told us that the hippocampus is needed for the formation and initial consolidation of memories, but not for storing those that have already been fully consolidated and transferred to the cortex.
Second, and this became one of the greatest discoveries of 20th century neuropsychology, some kinds of new learning still worked in Henry. The mirror drawing experiment was only the beginning. Milner and other researchers showed that Henry could improve his performance on a whole range of tasks requiring motor skills. He could learn to read mirror reversed text. He got faster and more accurate with every session. Yet every time he was given the task, he insisted he had never done it before. Henry also demonstrated perceptual priming, a form of implicit memory in which a prior encounter with information makes subsequent processing easier, even when the person does not remember the encounter itself. If Henry was shown an incomplete picture of an object and asked to guess what it was, and then, sometime later, was shown the same object again, he recognized it faster the second time, even though he did not remember seeing it before.
His brain processed repeated information more efficiently, even when his conscious mind registered no repetition. All of this led Squire to formulate one of the most important classifications in the science of memory. Squire divided memory into two broad categories. Declarative or explicit memory is what you can put into words. Episodic, I remember going to the store yesterday. Semantic, I know that Paris is the capital of France. These kinds of memory depend on the hippocampus for their formation and initial consolidation. These are what Henry lost. Non-declarative or implicit memory
is what is expressed through action rather than words. Procedural memory, how to ride a bicycle. Priming, faster processing of familiar stimuli. Classical conditioning, reflexes built through repetition. These kinds of memory depend on other brain structures, the cerebellum, the basal ganglia, the neocortex, and they remain intact when the hippocampus is damaged. Squire's classification became the canonical framework for all of memory neuroscience. It showed that the brain stores different kinds of information in different systems, and those systems can operate independently of one another. Henry Mollison proved this every day to everyone who worked with him. And here is the puzzle this created. If procedural memory forms without the hippocampus, if Henry could learn to draw in a mirror without remembering he had ever done it, then somewhere in his brain, changes were happening. Synapses were being strengthened somewhere. Neural circuits were being rewired somewhere. Learning was taking place. It was just taking place in systems that were not accessible to conscious recall. The body remembered what the mind did not. This dissociation between conscious and unconscious memory is one of the deepest lessons of neuroscience. It shows that consciousness, your feeling of "I remember," is not a required component of learning. The brain can learn, change, and adapt without you noticing. And the greater part of what your brain knows, skills, habits, automatic reactions, perceptual patterns, lives beyond the reach of your conscious awareness. Michael Kahana, who works with patients with implanted electrodes and studies the fluctuations of memory, puts it this way: the most interesting things that the brain does are the things it is not consciously aware of. All that stuff working behind the veil of consciousness, in the background, that may be affecting future behavior without us even suspecting it. Georgi Bousaki of New York University goes even further. What is me? He asks. Me is my memories. If you take away my memories, will I be aware of my surroundings? Perhaps. But will I be the same Bousaki I was 5 minutes ago? Without the past, you cannot have the kind of consciousness that philosophers talk about.
Squire's classification had profound implications that continue to ripple through neuroscience. Within declarative memory itself, there is a further division. Semantic memory, facts and world knowledge, gradually becomes independent of the hippocampus over time. When you first learn something, say that the capital of Australia is Canberra, that knowledge initially depends on the hippocampus. But over weeks and months, it gradually detaches from the specific episode in which you acquired it and becomes part of your general knowledge stored in the cortex, including the anterior temporal lobes. You know the fact, but you do not remember when or where you learned it. The fact has become semantic, free of episodic context. This transition from episodic to semantic happens through systems consolidation, the gradual transfer of information from the hippocampus to the cortex. And this is precisely why older adults can have excellent recall of facts and knowledge accumulated over a lifetime, but struggle to remember new events. Their cortex is rich with consolidated knowledge, but their hippocampus, needed for initial processing of new episodes, works less efficiently. Squire and Candle, in their co-authored book, Memory from Mind to Molecules, emphasize that the multiplicity of memory systems is not an accident or a design flaw. It is the product of evolution. Different kinds of memory arose at different times, in different evolutionary lineages, for different purposes. Procedural memory, memory for skills and habits, depends on circuits in the basal ganglia and cerebellum that are evolutionarily older than the hippocampus. Fish, reptiles, and birds all have it. Emotional memory, linked to the amygdala, is also present in all vertebrates, and connected to ancient circuits for evaluating threat and safety.
Declarative memory, dependent on the hippocampus, is a more recent evolutionary addition that reaches its greatest complexity in primates, and especially in humans. Your brain is not a single memory system elegantly designed from scratch. It is a geological column, layer upon layer of new systems built on top of older ones, each with its own history. And all of them run simultaneously, sometimes cooperating, sometimes competing, creating the rich, multi-layered experience we call my life. The hippocampus turned out not to be a storage vault for memory. It turned out to be something subtler and more important. The neuropsychologist Veronica O'Carroll, who spent decades working with patients with memory impairments, calls it the memory maker. But an even more precise term would be dispatcher or indexer. The hippocampus does not store memories whole, like files on a hard drive. Instead, it stores something like an index card, a pointer that knows which fragments of a memory are located in which parts of the cortex. The visual part of an episode sits in the visual cortex, the auditory part in the auditory cortex, the emotional part in the amygdala, the smell in the olfactory cortex. The hippocampus binds all of those fragments into a single experience. It is what makes a memory whole. When you remember, the hippocampus initiates a process neuroscientists call pattern completion. You receive a small fragment, a smell, a melody, someone's face, and the hippocampus uses that fragment as a search key. It activates neural connections leading to the remaining components of the memory, and gradually the full picture reconstitutes itself. Image, sound, smell, sensation, emotion. But it does not reconstitute completely. Oliver Baumann, a neuroscientist at Bond University in Australia, who studies neuroimaging of memory, explains it this way. When you see a new scene, a particular set of neurons in your visual cortex activates. When you remember that scene tomorrow, some of the same neurons in the same visual cortex will activate again, maybe 20%. It is not a full reproduction. It is a skeletal version, a scaffold. And that is precisely why a memory is never as vivid as the original experience. You do not put on virtual reality goggles when you remember. You see something like a pale, slightly blurred copy that nevertheless feels real. But right next to the hippocampus, literally adjacent to it, anatomically inseparable, sits another structure without which the story of memory would be entirely different. This is the amygdala, a small, almond-shaped nucleus that O'Carroll calls the emotional spark plug of the brain.
The amygdala and the hippocampus are so densely connected by neurons and dendrites that their wiring is virtually impossible to untangle. They work in tandem. The hippocampus creates the memory, the amygdala colors it with emotion. That is why emotional memories are brighter and more durable than neutral ones. When something triggers a strong emotion in you, fear, joy, surprise, grief, the amygdala sends a signal to the hippocampus. Remember this. Remember it firmly. And the hippocampus obeys. O'Carroll describes the cascade that fires when you encounter something emotionally charged. Imagine, a gun is pointed at your head. The light waves from the image of the gun hit your retina. An electrical signal races along the optic nerve to the back of your brain, to the visual cortex, where the image is recognized. Then the signal shoots inward, to the hippocampus. This is a threat, remember it. And simultaneously to the amygdala. This is danger, react. The amygdala triggers a cascade through the hypothalamus, the tiny structure that governs the autonomic nervous system. Your heart accelerates, adrenaline floods your blood, muscles tense, your entire body switches into fight or flight mode. And if the threat is severe enough, 10 to 20 minutes later, a second, slower mechanism kicks in. The hypothalamus sends a signal through the bloodstream to the adrenal glands, which begin producing cortisol, the stress hormone that maintains a state of vigilance for hours. That entire cascade, from light wave to cortisol, serves a single purpose: to make sure you never forget this moment. Evolution ensured that experience threatening your life is recorded as firmly as possible. Because if you survive, you will need that recording. Next time, you will react faster. But this mechanism has a dark side.
If stress becomes chronic, in depression, in post-traumatic stress disorder, in constant anxiety, cortisol, which was supposed to be a short-term helper, becomes a destroyer. Chronically elevated cortisol levels literally shrink the hippocampus. The dendrites of its neurons, those branching extensions that receive signals from other neurons, begin to retract. The hippocampus loses volume, and simultaneously, the amygdala does the exact opposite. It expands, its neural connections growing denser and more powerful. The result is a trap. The longer a person remains in a state of stress, the weaker their memory becomes, and the stronger their emotional reactions grow. Weakened memory cannot effectively contextualize fear, and the amplified amygdala generates ever more anxiety, which produces even more cortisol, which shrinks the hippocampus further. This is what is called the cascade hypothesis, a vicious cycle that is very hard to escape without outside help.
Helen Mayberg, a neuroscientist at the Icahn School of Medicine at Mount Sinai in New York, who spent 15 years studying the neural circuits of depression, describes depression as a state in which the brain is locked. She identified a specific region, the subcallosal cingulate, which she says acts as the ringleader in this locked circuit. Its connections to the thalamus, the hippocampus, the frontal cortex, the hypothalamus. If this node is not unlocked, everything else remains stuck. Mayberg began using deep brain stimulation, implanted electrodes delivering high-frequency pulses to this region for patients with severe, treatment-resistant depression. People who had not responded to medication, psychotherapy, or electroconvulsive therapy. People who were, essentially, at the end of the line. And what happened in the operating room was remarkable. When the electrodes began delivering stimulation, patients described it this way: the negative just switches off.
It is as though I am reconnecting to the world. I can move. I can feel. I can pay attention to what is around me. The paralysis of depression retreated within minutes. And if stimulation continued in the operating room for 10 to 15 minutes, the effect lasted for weeks, even without further stimulation. Something was resetting in the system. Then, with continuous stimulation through an implanted neurostimulator, patients gradually, over months, regained the ability to interact with the world, rebuilding their internal map of reality. One of Mayberg's patients, coming in for a 10-year follow-up, described her recovery process in exactly the terms proposed by the neuroscientist Gheorghe Buzaki: updating the internal model of the world through action. Buzaki has spent years promoting the idea he calls the inside-out view. The brain is not a passive receiver of information from the outside, but a self-organizing system that builds a model of the world through its own actions and continuously updates that model by comparing predictions with results. When this ability to update the model breaks down, as in depression, the person gets stuck. They cannot relearn. They cannot correct their picture of the world. And that is what makes depression so destructive to memory. It is not just the storage that suffers. It is the entire updating mechanism. Oliver Baumann, the neuroscientist from Bond University, whose work on spatial perception and memory we already mentioned, explains the relationship between emotion and memory through the metaphor of investment. Encoding a memory is an expensive process for the brain, synthesizing new proteins, restructuring synapses, transporting receptors. All of it requires energy, and the brain already consumes roughly 20% of the body's total energy, despite making up only about 2% of its weight. The organism cannot afford to encode every waking moment with equal intensity. It needs a signal that says, "This one matters. Invest resources." Emotion is that signal. When something provokes an emotion in you, any emotion, positive or negative, it means your brain has assessed the situation as significant for survival, well-being, or social standing. Dopamine, serotonin, norepinephrine, released during emotionally charged events, act as molecular markers of importance. They tell the hippocampus and the amygdala, this moment is worth remembering. And the synaptic machinery responds accordingly, investing more resources in consolidating that particular episode. That is why you remember your first kiss, but not lunch on a Tuesday 3 weeks ago.
The first kiss was emotionally loaded. Excitement, anticipation, maybe fear. And all those emotions tagged it as significant. Tuesday's lunch, if nothing special happened during it, received no tag, and the brain rationally decided not to spend resources on its consolidation. There, by the way, is part of the answer to one of our big questions: why we remember some things and forget others. Not because some events are more important in some abstract sense, but because some events were accompanied by a release of neurotransmitters that triggered enhanced consolidation, and others were not. Your brain does not decide what to remember based on rational assessment. It responds to a neurochemical signal. And that signal is tied to emotion. Gail Robinson, the neuropsychologist, who will later tell us about people with exceptional autobiographical memory, notes that when we actively pay attention to something, when we consciously focus, that also strengthens encoding. The prefrontal cortex, responsible for attention and control, sends top-down signals to the hippocampus, facilitating the formation of a memory. That is why mindfulness improves memory. If you are truly present in the moment, if your attention is fully directed at what is happening, you give the brain a second channel of tagging beyond emotion. Robinson draws a practical conclusion. When someone complains that they are constantly losing their keys, forgetting names, and cannot remember where they put their phone, the first question she asks is, "What were you thinking about at that moment? Where was your mind?" And almost always the answer is, "I was thinking about something else." The keys were set down on autopilot while the mind was occupied with tomorrow's meeting or a grocery list. Attention was pointed elsewhere. The brain did its job honestly. It did not spend resources encoding a moment that you yourself flagged as insignificant through your inattention.
Edward Chang, a neurosurgeon at the University of California, San Francisco, who works on decoding the neural signals of speech, describes an analogy that helps grasp how the brain encodes memories through the lens of another, better-studied function: speech. Chang and his team spent a decade deciphering the neural code for speech, the patterns of electrical activity in the motor and sensory cortex that encode individual sounds, syllables, and words. Working with patients who had electrodes implanted on the surface of their brain for epilepsy treatment, Chang mapped the zones responsible for each element of speech. One zone, roughly a millimeter across, controls the movement of the tongue tip for the D sound. The adjacent zone controls the lips for the B sound, and so on, a complete map of the vocal tract on the surface of the cortex. In English, there are only about 40 phonemes, the elementary sounds from which all words are built. In just a few minutes of ordinary conversation, you cycle through that entire dictionary more than once. Every new sentence is a recombination of those same 40 elements in a new sequence to express a new meaning. A finite set of units, an infinite number of combinations. Memory works on a similar principle, except the dictionary is immeasurably larger, and the units of combination are not phonemes but neural ensembles. Every new memory is a new combination of neurons, some of which participate in many other memories. It is this combinatorics that explains how a brain with a finite number of neurons can store a practically unlimited number of memories. Not by creating new neurons for every new event, but by creating new combinations of existing ones. Michael Halassa from MIT, who studies the mechanisms of cognitive control and attention, emphasizes that one of the brain's chief tasks is filtering. We are constantly flooded with information.
Electromagnetic waves striking the retina, sound waves pressing on the eardrum, tactile sensations from clothing and surfaces. The volume of incoming data is staggering. If the brain tried to process all of it equally, it would be paralyzed. Halassa discovered that filtering happens through circuits between the prefrontal cortex and a structure called the mediodorsal thalamus. These circuits determine which information gets priority, which gets filtered out, and which memories are activated for the current task. The prefrontal cortex sends what are called top-down instructions to sensory areas and to the hippocampus. Amplify the signal on this channel. Suppress the noise on that one. Retrieve this memory. These instructions determine what you pay attention to and, consequently, what you will remember.
Halassa describes it as a system of gatekeepers. Information is trying to break through to attention and awareness, and the gatekeepers decide what to let in and what to leave outside. And these gatekeepers operate not only in the realm of sensation, but in the realm of memory and thought as well. Even when you are trying to recall something, the search process is filtered. From among many potentially relevant memories, the gatekeepers select those most likely to be useful for the current task. Now we know the map. We know that the hippocampus is the dispatcher that binds memory fragments together. We know the amygdala adds emotional weight. We know the cortex stores the fragments, visual, auditory, tactile, olfactory. We know the prefrontal cortex manages attention and working memory, and we know that damage to these structures breaks memory in predictable and specific ways. But all of this is the top-level map. We know which buildings stand in the city. We know which streets connect them. But we have not yet looked inside any building. We have not seen what happens at the level of connections between individual neurons, at the level of synapses, those tiny gaps across which one nerve cell transmits a signal to another. And it is there, at that level, that the real story of how experience becomes memory begins. Because if the hippocampus is the dispatcher, then the synapses are the actual records. And to understand how they work, we need to go down one floor. And for that, we need to travel to 1949, to Montreal, to a man who guessed the answer 20 years before anyone could test it. His name was Donald Hebb, and his idea was so simple that at first many people refused to believe it could be right. Donald Hebb was a Canadian psychologist, quiet, methodical, the kind of person who does not give the impression of a visionary, but who changes the direction of an entire scientific discipline with a single paragraph. In 1949, he published a book called The Organization of Behavior, and in that book, he formulated a principle that has since become one of the most cited statements in neuroscience. In his original wording, it is rather verbose, but the idea extracted from it fits in 5 words. Neurons that fire together, wire together. That is not a direct quote from Hebb, it is a later paraphrase of his idea, but it captures the essence precisely. Imagine 2 neurons, A and B. Neuron A sends a signal to neuron B. If neuron A is active at the very moment when neuron B is also active, if they fire at the same time, the synaptic connection between them strengthens. They begin to hear each other better. The next time neuron A fires, it will be easier for it to trigger neuron B. And conversely, if two neurons are never active simultaneously, their connection weakens. This sounds simple, almost obvious, but think about what it means.
It means that the very act of two neurons being active together changes the physical structure of the connection between them. Experience, literally the electrical activity caused by seeing, hearing, or feeling something, leaves a physical trace in nerve tissue. Hebb proposed that this is how learning works. This is how experience becomes memory. Not through some mysterious immaterial recording, but through concrete, measurable, physical changes in the connections between nerve cells. The problem was that Hebb proposed this in 1949, and he had no way to prove it. The technology of the time could not measure changes in the strength of an individual synapse. Hebb advanced his hypothesis on the basis of logical reasoning and indirect evidence, and he had to wait nearly a quarter of a century before anyone found direct confirmation. But while neurophysiologists in Europe and America were unsuccessfully trying to find Hebbian plasticity in the mammalian brain, a young scientist at the other end of the spectrum of biological complexity was approaching the same problem from an entirely different direction. His name was Eric Kandel, and his solution turned out to be simultaneously brilliant and absurdly simple. If the mammalian brain is too complex for studying memory at the level of individual synapses, find a simpler animal. Kandel was born in 1929 in Vienna into a Jewish family. He was 9 years old when the Anschluss happened, the annexation of Austria by Nazi Germany. His family fled to America. Kandel later said that the early experience of loss, the loss of home, the loss of a world he knew, shaped his interest in memory. How does the human brain store the past? How do we carry inside us something that no longer exists?
He started as a psychiatrist, planning to work with patients, to explore the unconscious in the Freudian tradition. But then he decided that if he wanted to understand how the brain stores experience, he needed to go down to the level of individual cells. And for that he needed a simple animal. So simple that you could see every neuron, record every signal, trace every connection. Kandel found Aplysia californica, the California sea hare, a giant sea slug that lives off the coast of California. Aplysia is a creature roughly a foot long, soft, slow, utterly homely. But it had one invaluable quality. Its nervous system contained only about 20,000 neurons, many of which were so large that they could be seen with the naked eye, and their electrical signals could be recorded individually. For comparison, the human brain contains about 86 billion neurons. Aplysia has 20,000, and those 20,000 neurons were enough for the animal to learn.
Kandel focused on the simplest reflex Aplysia has, the gill withdrawal reflex. The slug has a fleshy protrusion called a siphon, and next to it, a delicate gill. If you touch the siphon, the slug retracts the gill, a simple protective reflex. A sensory neuron in the siphon detects the touch, sends a signal to a motor neuron. The motor neuron activates a muscle. The gill pulls in. 3 neurons, 1 synapse, 1 reflex. And here is what Kandel showed. If you touch the siphon again and again, gently, harmlessly, the slug gradually stops retracting its gill. It habituates. This is habituation, the simplest form of learning.
The slug learns that the touch is not dangerous and stops responding to it. And this learning happens through a specific, measurable change in the synapse between the sensory and motor neurons. The mechanism turned out to involve the calcium channels on the presynaptic terminal.
With repeated stimulation, these channels begin admitting less calcium, and calcium is necessary for the synaptic vesicles containing neurotransmitter to fuse with the membrane and release their contents. Less calcium, fewer vesicles fuse, less glutamate released, weaker response from the motor neuron, weaker gill retraction. The synapse weakens. The connection gets quieter. Habituation. The reverse also worked. If instead of a gentle touch, the slug received a painful stimulus, a mild electric shock to the tail, and then another gentle touch to the siphon, the gill withdrawal response became stronger than it had been before the shock. The slug learned that there was danger in the environment, and its reflex sharpened. This is sensitization. And here, a third neuron entered the game, a modulatory neuron. This neuron, activated by the tail shock, released serotonin onto the terminal of the sensory neuron. Serotonin did not activate the sensory neuron directly.
Instead, it triggered an intracellular cascade, each step of which Kandel and his colleagues traced with near-surgical precision. Serotonin bound to a receptor on the sensory neuron's membrane. The receptor activated the enzyme adenylyl cyclase, which produced cyclic AMP, a critical second messenger. Cyclic AMP activated protein kinase A, which phosphorylated potassium channels in the membrane, delaying their closure. This lengthened the action potential in the sensory neuron, which in turn increased calcium influx, which increased glutamate release. Result? The next touch to the siphon would produce a stronger response. The synapse got louder. For short-term sensitization, lasting minutes, this was enough. Existing proteins were modified, ion channels were retuned, the synapse worked louder for a while, and then everything returned to baseline. Nothing new was built. The existing machinery simply worked a little differently.
But long-term memory, sensitization lasting days and weeks, required something fundamentally different. Kandel found that if you delivered the shocks repeatedly, at specific intervals, sensitization became long-lasting. And for that, new proteins were necessary. Repeated serotonin exposure from multiple tail shocks raised cyclic AMP and protein kinase A levels high enough that the catalytic subunit of protein kinase A migrated from the cytoplasm into the cell nucleus. In the nucleus, it phosphorylated CREB, the transcription factor that binds to DNA and controls gene expression. CREB activated genes. Protein synthesis began, and these new proteins built new synaptic contacts. Kandel and his colleagues Craig Bailey and Mary Chen demonstrated this anatomically. After long-term sensitization, the number of synaptic endings between the sensory and motor neurons increased. Physically, you could count them.
Where before learning there had been, say, 1,300 synaptic contacts, after learning there were about 2,700. Memory was literally building new matter. And when long-term sensitization was extinguished, when the slug stopped receiving shocks, the number of synaptic contacts returned to baseline. Synapses built by learning were dismantled when the learning was no longer relevant. Anatomy followed behavior. Kandel described it this way. Short-term memory is a functional change, a modification of existing connections. Long-term memory is a structural change, the building of new connections. The switch between the first and the second is CREB. If CREB is activated, genes turn on, proteins are synthesized, synapses are built, and memory becomes long-term. If CREB is not activated, none of that happens, and the memory dissipates. For this work, Kandel received the Nobel Prize in Physiology or Medicine in 2000.
But let us pause before we move on, because this is important. When we talk about serotonin triggering a cascade in the neuron of a sea slug, it might seem as though we have wandered off topic. A sea slug. A gill withdrawal reflex. What does any of this have to do with your memory of your grandmother's kitchen? Everything. The molecular language that Kandel deciphered in Aplysia, serotonin, cyclic AMP, protein kinase A, CREB, synthesis of new proteins, building of new synapses, is the very same language your brain is using right now to record this evening. Evolution did not bother to invent a new mechanism for each new species. It found a working solution 600 million years ago in the common ancestor of worms, slugs, and humans, and since then it has only modified, elaborated, and built on top of it. Your hippocampus is, in essence, an extraordinarily complex version of that simple synapse in Aplysia. The molecules are the same, the principles are the same.
The difference is in scale, in the number of neurons, in the complexity of the networks, in the added layers of regulation. But the foundation remains. While Kandel was working with Aplysia on the Atlantic coast of America, across the ocean, something equally important was happening. In 1973, two researchers, Tim Bliss and Terje Lomo, working in Per Anderson's laboratory in Oslo, Norway, discovered a phenomenon that became the gold standard for studying synaptic plasticity in vertebrates. They were working with the rabbit brain, specifically with the hippocampus. They stimulated the perforant path, one of the major inputs to the hippocampus, with a short, high-frequency burst of electrical impulses, a few seconds of rapid stimulation. And then they measured how hippocampal neurons responded to ordinary single stimuli after that burst. The response was stronger, significantly stronger. The synapses that had received that high-frequency stimulation became more sensitive. They responded to a standard signal with greater amplitude. And, this is the key, the enhancement did not fade after a few minutes. It held for hours, for days, in some experiments, for weeks. Bliss and Lomo called this phenomenon long-term potentiation, or LTP, and it became the cellular correlate of learning. Here is what happens at the level of the synapse when you learn something. The synaptic connection strengthens, and that strengthening can last a very long time. Hebb was right. Neurons that are active at the same time really do strengthen the connection between them. LTP is the molecular implementation of Hebb's rule. But how exactly does it happen? What mechanism allows a synapse to remember that it was stimulated? The answer turned out to involve one particular receptor, and this receptor works as a molecular coincidence detector. It is called the NMDA receptor, and its properties correspond so elegantly to Hebb's rule that when scientists first described them, some physiologists simply could not believe that evolution could have created something so precisely suited to the task. The NMDA receptor is an ion channel in the membrane of the postsynaptic neuron, the neuron receiving the signal. It responds to glutamate, the main excitatory neurotransmitter in the brain. But the NMDA receptor has a feature that distinguishes it from all other glutamate receptors. At rest, its channel is blocked by a magnesium ion. The magnesium sits in the channel like a plug, preventing other ions from passing through even when glutamate has bound to the receptor. To remove that magnesium plug, the postsynaptic membrane must be depolarized, meaning the postsynaptic neuron must already be active.
Think about what this means. The NMDA receptor opens only when two conditions are met simultaneously. The presynaptic neuron has released glutamate, meaning the presynaptic neuron is active, and the postsynaptic neuron is depolarized, meaning it too is active. Both neurons must be active at the same moment. If only one is active, nothing happens. The NMDA receptor requires coincidence. It is the molecular embodiment of Hebb's rule. When both conditions are met and the NMDA receptor opens, calcium ions flood into the postsynaptic neuron. And calcium is the signal. Calcium triggers an entire cascade of molecular events inside the cell that ultimately leads to the strengthening of the synapse. And here we descend one more floor, to the molecules themselves that make the strengthening possible. Because calcium is only the trigger,
What happens after it is an intricate biochemical ballet that unfolds from milliseconds to hours, from the cell surface to its nucleus and back. And it is this ballet that transforms a fleeting experience into a lasting memory. There is one more observation that connects the molecular level of memory with the level of the whole organism, and it has to do with attention and interest. You have probably noticed that learning something new is easier when you are interested. When a topic grabs you, when you are engaged, when you genuinely care, information seems to settle into your head on its own. But when you are bored, when you are forced to cram something that does not move you, every fact has to be hammered in by force, and it falls out again within a day. At the molecular level, this is explained by the fact that engagement and interest are accompanied by a release of dopamine, the neurotransmitter associated with motivation and reward.
Dopamine, like serotonin and norepinephrine, modulates synaptic plasticity. It enhances LTP in the hippocampus, facilitates CREB activation, and promotes more efficient consolidation. When you are interested, your synapses literally find it easier to learn. This creates a positive feedback loop. The more you know about a topic, the easier it is to integrate new information, because you already have neural networks to which it can attach. The more pleasure you get from learning, the more dopamine is released, the more efficiently synaptic plasticity works, and the more easily you remember. This is why experts in their field absorb new material faster than beginners. Their brains are already tuned to the subject, already have a web of connections, and every new fact does not have to be written from scratch. It plugs into the existing architecture. The reverse is also true. Boredom is a signal to the brain. Nothing new here. Do not spend resources. Dopamine is not released.
LTP is not boosted. Consolidation proceeds sluggishly. Information does not stick. That is why cramming is the least efficient method of learning. It lacks the emotional and dopaminergic signal that tells the brain, remember this. And here is yet another small answer to the big question from the beginning of our journey. Why do you remember the smell of tangerines from childhood, but not lunch on Tuesday? Because the tangerines were bound up with warmth, with people you loved, with celebration. And that emotional cocktail switched on every molecular amplifier of consolidation. Tuesday's lunch passed on autopilot, without emotion, without dopamine, without tagging. Your brain did its job honestly. It kept the important, and it released the insignificant. Let us follow the calcium and see where it takes us. So the NMDA receptor has opened, and calcium has flooded into the postsynaptic neuron. What happens next? Calcium is one of the most versatile signaling ions in biology. Cells use it for a wide range of purposes, from muscle contraction to cell division, from hormone secretion to triggering apoptosis, programmed cell death. But in a hippocampal postsynaptic neuron, calcium arriving through the NMDA receptor does something very specific. First, it binds to a protein called calmodulin. Calmodulin is a small intermediary protein that changes shape when calcium ions attach to it, and in this new shape, it can activate other proteins. Specifically, the calcium-calmodulin complex activates an enzyme called CAMK2, calcium-calmodulin-dependent protein kinase type 2. The name is long, but this enzyme is worth remembering because it may be one of the key molecular carriers of short-term memory. CAMK2 does something remarkable. It phosphorylates other proteins. Phosphorylation is the attachment of a phosphate group to a protein, which changes its shape and, consequently, its function. And one of the proteins that CAMK2 phosphorylates is the AMPA receptor, another glutamate receptor on the surface of that same synapse. A phosphorylated AMPA receptor admits more sodium ions each time it opens. The synapse becomes louder. The same signal from the presynaptic neuron produces a stronger response in the postsynaptic one. But that is not all. CAMK2 also initiates a process in which additional AMPA receptors, previously stored inside the cell in special vesicles, are transported to the membrane surface and inserted into it. Now there are more AMPA receptors at the synapse than before. More receptors, more sodium ions with each signal, a stronger response. The synapse has become more sensitive. It remembers that it was activated. All of this happens in the first minutes after learning, and it does not require the synthesis of new proteins. The cell uses components it already has, simply reshuffling them, moving them from one location to another, modifying them.
That is why you can hold a phone number in your head for a few minutes without any effort. Your synapses do not need to build anything new. They just slightly retune the existing machinery. But this retuning is short-lived. The phosphate groups fall off. The extra AMPA receptors are gradually pulled back inside the cell. Without reinforcement, the synapse will return to its original state in an hour or two. Short-term memory is, in essence, written in pencil that erases itself. For a memory to last days, weeks, years, for long-term memory, the next stage is necessary. And this stage requires something that short-term memory does not. It requires the neuron to synthesize new proteins. And to synthesize proteins, certain genes in the cell's nucleus must be switched on. Here is how it works. The calcium signal that began at the synapse propagates inward through the cell.
Through a series of intermediaries, it reaches the neuron's nucleus, where it activates the transcription factor we have already discussed, CREB. Activated CREB binds to the promoter regions of specific genes and switches them on. The cell begins producing mRNA, messenger ribonucleic acid, the intermediary molecule that carries instructions from the gene to the ribosomes, the molecular factories that assemble proteins. And the ribosomes begin building new proteins. Among these proteins, one deserves special attention, ARC, activity-regulated cytoskeleton-associated protein. ARC is the master regulator of synaptic plasticity. It controls the number of AMPA receptors at the synapse, their trafficking, their distribution. When ARC is synthesized in neurons that were active during learning, it locks in the changes produced at the previous stage. The extra AMPA receptors embedded in the membrane are now held in place. The synapse stabilizes in its strengthened state.
But structural changes go even further. Over the hours and days following learning, dendritic spines, tiny protrusions on the surface of dendrites, each containing a single synapse, change shape. They grow larger. Their internal cytoskeleton the scaffolding of protein filaments that gives the spine its form, is remodeled. New cytoskeletal proteins, actin, drebrin, and others build a bigger, sturdier structure. The synapse that was strengthened by learning becomes physically larger and physically stronger than a synapse that was not involved in learning. Here is what this means. A memory is not an abstraction. It is not a vague change in the brain. When you learn something, when you remember something, specific synapses in specific neurons of your brain physically change. The number of receptors on their surface changes. The shape of dendritic spines changes. The molecular composition changes. New proteins are synthesized and incorporated into the structure. If you could compare a synapse before and after learning under an electron microscope, you would see the difference. Memory is a concrete, physical material change in the tissue of your brain. CAMK2 has yet another remarkable property that makes it a candidate for the role of molecular custodian of short-term memory. When CAMK2 is activated by calcium, it can autophosphorylate, that is, phosphorylate itself. And in this autophosphorylated state, it remains active even after the calcium is gone. It is as though it remembers being activated. This is a molecular switch. Once turned on, it stays on for a while, regardless of whether the incoming signal continues. Some researchers propose that autophosphorylated CAMK2 is the molecular substrate of short-term memory, the mechanism that holds information for minutes, until either long-term consolidation is triggered or the information is lost. But from short-term to long-term memory is a qualitative leap, and that leap is defined by a single, fundamental question. Will the signal reach the nucleus? If the calcium cascade is strong enough and sustained enough, if the experience was significant enough, repeated enough, emotionally loaded enough, the signal crosses a threshold and activates transcription factors in the nucleus. CREB switches on. Genes are read. Proteins are built. The synapse goes from temporarily strengthened to structurally remodeled. Memory transitions from an unstable state to a stable one. This transition is a moment of vulnerability. If you block protein synthesis within roughly 6 hours of learning, long-term memory does not form. The experience remains in short-term memory and gradually fades. This is the consolidation window, the period during which the memory has not yet stabilized and can be lost.
It is precisely in this window that sleep intervenes. During slow-wave sleep, the hippocampus replays fresh memories, reinforcing and solidifying the consolidation process. One more protein that plays a critical role in the structural changes of long-term strengthening deserves mention. BDNF, brain-derived neurotrophic factor, a growth protein for neurons. BDNF promotes the growth and branching of dendrites, enhances formation of new synapses, and supports neuron survival. Its expression increases with learning, and blocking BDNF impairs long-term memory formation. BDNF is, if you will, the construction material that CREB orders by switching on genes. Without BDNF, the order remains on paper. Genes are on, but the building of new synaptic contacts stalls. Dendritic spines, which we mentioned, are not passive structures. They are active, dynamic formations that are constantly appearing, disappearing, and changing shape. In the brain of a healthy adult, they exist in a state of continuous renewal. Learning stabilizes certain spines, making them larger and more robust, and at the same time can cause others to disappear. Memory does not simply add new structures. It remodels the existing architecture of synapses, strengthening some connections and weakening others. There is a question that often puzzles people. If memory is stored in proteins, and proteins in a cell are constantly being broken down and replaced, the average protein lifespan in a neuron is measured in days or weeks. Then how can a memory last for years? The proteins that held it yesterday have already degraded and been replaced by new ones. How does memory survive the renewal of its own substrate? Think of a river. The water in a river is continuously renewed. The water molecules passing through a given stretch yesterday are long gone, already on their way to the sea. But the river remains a river.
Its shape, its bed, its bends, its banks, all of that is stable, even though the substrate, the water, is continuously renewed. The shape of the river is determined not by the water molecules, but by the landscape that guides their flow. So it is with memory. The proteins at the synapse are the water. They come and go. But the epigenetic code on the neuron's chromatin is the landscape, the riverbed. It determines which proteins will be synthesized, which receptors will be embedded in the membrane, what shape the dendritic spine will take. As long as the landscape is stable, the river flows along the same bed, even if every drop in it is new. As long as the epigenetic code on the promoter of the ARC gene is preserved, the neuron will resynthesize the necessary proteins, and the synaptic change encoding the memory will be maintained, even if every individual protein molecule at the synapse has been replaced dozens of times since the original learning event.
Here is the answer to the puzzle. Memory survives protein renewal not because the proteins are permanent, they are not, but because the instructions for producing them are stored on a more stable medium, on the chromatin, in the epigenetic code, which is reproduced by enzymes and maintained by molecular proofreaders. It is as though you had a recipe for a cake. The cake can be eaten and a new one baked from the same recipe, and it will be the same. The recipe outlives the cake. The answer to this question was for a long time one of the hardest riddles in neurobiology. But in recent years, it has begun to come into focus, and for that we need to go down one more floor, from proteins to DNA, from the cytoplasm to the nucleus, from the synapse to the chromatin. Because, as it turns out, the deepest layer of memory is not the changes on the surface of the neuron. The deepest layer is changes in how the neuron reads its own genes. And this level has proven to be the key not only to the persistence of memories, but to a possibility that 10 years ago seemed like pure science fiction, switching specific memories on and off like a toggle. Let us take a moment to go back to how DNA is organized, because without this, the next part of the story will not make sense. You probably know that DNA is a double helix, a long molecule twisted around its own axis. Every cell in your body contains a complete copy of your genome, roughly 3 billion base pairs, about 20,000 genes. If you stretched out the DNA from a single cell into a line, it would be roughly 6.5 feet long. But the cell nucleus, where that DNA is housed, has a diameter of about 6 micrometers, roughly 3,000 times smaller than the length of the molecule. To fit 6.5 feet of DNA into such a tiny space, the molecule must be packed incredibly tightly. The packing looks like this. DNA is wound around small protein spools called histones. About 146 base pairs of DNA are wrapped around an octamer of 8 histone proteins, forming a nucleosome, the basic unit of chromatin packaging. Nucleosomes are strung along the DNA like beads on a string, and then that string of beads coils further, forming a chromatin fiber, which twists and folds into even more compact structures. And here is what matters. The degree of packing directly affects whether a gene is accessible for reading or not. Where chromatin is tightly wound and condensed, a state called heterochromatin, the molecular machinery that reads genes simply cannot reach the DNA. The gene is locked. It is silent. Where chromatin is loosened and open, euchromatin, the DNA is accessible and the gene can be read.
The state of chromatin, tight or loose, is regulated by chemical modifications, small molecular tags that attach either to the DNA itself or to the histones. DNA methylation, the attachment of a methyl group to cytosine, one of the 4 DNA bases, generally leads to chromatin compaction and gene silencing. Histone acetylation, the attachment of an acetyl group to the tails of histone proteins that protrude from the nucleosome, generally loosens the bond between DNA and histone, opens the chromatin, and allows the gene to work. Histone methylation can go either way, depending on which specific amino acid residue is modified. Trimethylation of lysine 4 on histone H3, H3K4ME3, typically activates a gene, while trimethylation of lysine 27, H3K27ME3, typically silences it. These chemical tags are called epigenetic modifications, from the Greek epi, meaning above, as in above genetics. They do not change the sequence of letters in the DNA.
Your genes remain the same. But epigenetic tags determine which of those genes will be read and which will remain silent. Think of it as a dimmer on a lamp. The DNA is the bulb. It is the same bulb regardless, but the dimmer can make it bright, dim, or switch it off entirely. Epigenetic tags are the dimmers on your genes. Beyond methylation and acetylation, other epigenetic mechanisms participate in writing memory. One is chromatin remodeling, the physical repositioning of nucleosomes along the DNA. Special protein complexes called chromatin remodelers use the energy of ATP to slide nucleosomes, opening or closing stretches of DNA. You can picture this as sliding curtains on a window. The curtain is the nucleosome. The window is the stretch of DNA containing a gene. And whether the curtain is open or closed determines whether light, in our case the molecular reading machinery, reaches the gene. Another mechanism involves variant histones.
Not all nucleosomes are assembled from the same histone proteins. There are alternate histone variants that get incorporated into chromatin in place of the standard versions, changing the properties of the nucleosome, its stability, its accessibility to transcription factors, its interaction with neighboring nucleosomes. Swapping a standard histone for a variant is yet another way a cell can mark a specific region of the genome as special, requiring particular treatment. All of these mechanisms, DNA methylation, histone modifications, chromatin remodeling, variant histones, non-coding RNAs, work not in isolation but as an orchestrated ensemble. Each instrument adds its voice to the overall score, and the result of that score, the specific epigenetic state of a specific gene in a specific neuron, determines whether that neuron will produce the proteins needed to maintain a specific synaptic change, which is to say, ultimately, whether a memory will live. A study published in Science Advances in 2024 by Espinosa Martinez and colleagues described what the authors called the epigenetic cycle, a multi-phase system through which epigenetic memory marks are maintained during cell division. Although brain neurons mostly do not divide, except in zones of neurogenesis in the hippocampus, this mechanism is fundamentally important for understanding how memory is preserved at the organismal level and how it can be transmitted across cellular generations, in the immune system, in stem cells and, as we will see, across generations of organisms. At the center of the epigenetic cycle are two classes of chromatin modifications: DNA methylation, which is copied during replication by the enzyme DNMT1, which specifically recognizes hemimethylated DNA after division and restores the full methylation pattern on the new strand. And histone modifications, particularly H3K27ME3, catalyzed by polycomb repressive complex 2, which maintain the silenced state of developmental genes. These two systems do not merely lock in the current state of the cell. They actively restore it after every division, rewriting the instructions each time for which genes should be on and which should be off. A cell remembers what it is because its chromatin carries a record of that identity. And with every division, that record is copied and passed to daughter cells. Every time a liver cell divides and produces two liver cells, rather than a neuron and a muscle cell, that is an act of cellular memory, written in the epigenetic code and reproduced with extraordinary fidelity. Now let us return to memory. Tim Breedy, Professor of Cognitive Neuroepigenetics at the Queensland Brain Institute, studies exactly this. What happens to the DNA of neurons when you learn something? And what he has found shifts the story of memory to an entirely new level of depth. Brady explains it this way. Every time you experience an emotionally charged event, the brain releases neurotransmitters, dopamine, serotonin, norepinephrine. We already know that these molecules act on synapses, altering the strength of connections between neurons. But here is what we did not know until recently. These neurotransmitters also penetrate inside the neuron's nucleus. They reach the chromatin. And there they interact with histones, triggering a cascade of epigenetic modifications. DNA that had been tightly packed loosens up. Genes that were inaccessible become available for reading. Specific stretches of chromatin change their state. Methyl groups attach or detach. Histones are acetylated. The structure of the DNA can even temporarily shift from the right-handed helix into a left-handed form, the so-called zDNA, which makes it even more mobile.
All of this primes certain genes for expression and simultaneously marks that neuron as a participant in a specific learning episode. Among the genes activated during this process, the one playing the starring role is our old friend, ARC. The promoter of the ARC gene, the stretch of DNA that controls whether the gene will be read, undergoes specific epigenetic modifications in neurons that were active during learning. DNA methylation at the promoter changes. Histone acetylation around it changes. The chromatin at that location transitions to a more open state. And here is what makes this story truly deep. These epigenetic changes on the ARC promoter are not merely consequences of learning. They are instructions for the future. The next time you recall that experience, when the hippocampus initiates pattern completion and that neuron fires again, the ARC gene will already be in a more accessible state. It will switch on faster. ARC protein will be synthesized sooner.
Synaptic changes will be restored more efficiently. The neuron remembers that it participated in recording that experience because its chromatin carries the mark of that event. Epigenetic modifications are the answer to the question we asked a moment ago. How does memory survive protein renewal? Proteins at the synapse really do degrade and get replaced, but epigenetic tags on the chromatin are more stable. They are copied during certain processes, maintained by enzymes that check and refresh them. As long as the epigenetic tag on the ARC promoter endures, the neuron can resynthesize the proteins needed to maintain the synaptic change, even if the previous batch of those proteins has already broken down. The epigenetic code is a long-term instruction that outlasts the turnover of the protein substrate. But there is yet another element of this story that makes it even more extraordinary. Non-coding RNAs.
Reddy describes them as a new continent of neurobiology, territory we are only beginning to explore. When we think of genes, we usually think of them as instructions for building proteins. A gene encodes a protein. That is the classic central dogma of molecular biology. But it turns out that only about 2% of the human genome encodes proteins. 2%. The remaining 98% was long dismissed as junk DNA, useless baggage left over from evolutionary history. But in the last 2 decades, it has become clear that this is not junk at all. A large portion of that non-junk 98% produces RNA molecules that are not translated into proteins, but perform regulatory functions. They are called non-coding RNAs, and they have turned out to be critically important for memory. Brady and his colleagues discovered that certain classes of non-coding RNAs act as traffic managers inside the neuron. After learning triggers epigenetic changes in the nucleus and genes begin to be read, non-coding RNA molecules exit the nucleus and travel along the neuron's dendrite to the synaptic ending. There they coordinate the work of proteins that remodel the synapse, directing where to insert new receptors, how to restructure the cytoskeleton, which molecules to recruit for stabilizing the structure. In effect, non-coding RNAs are couriers, carrying instructions from headquarters, the cell nucleus, to the front line, the synapse. They ensure communication between the 2 levels of memory recording, the deep epigenetic level in the nucleus and the surface structural level at the synapse. Brady makes an observation worth holding on to. RNA, he says, is the most ancient information molecule on Earth. RNA appeared before DNA. In the RNA world hypothesis, the first forms of life used RNA both for storing genetic information and for catalyzing chemical reactions.
DNA and proteins came later as specialized tools. And now it turns out that RNA, this most ancient molecule, plays a central role in one of the most complex functions of the most complex organ, the formation and maintenance of memory in the brain. The molecule with which life began is still coordinating the process that makes us who we are. Brady describes the architecture of memory at the molecular level as a 2-layer system. On the surface level, synaptic changes, AMPA receptors, dendritic spines, structural remodeling. On the deep level, epigenetic modifications in the nucleus, DNA methylation, histone acetylation, non-coding RNA expression. The surface level enables rapid readout. When a memory is activated, synaptic changes determine which connections are louder and which patterns will be restored. The deep level provides the long-term instruction.
When proteins at the synapse degrade and are replaced, the epigenetic code in the nucleus endures and allows the neuron to resynthesize the needed proteins. It is like having a blueprint for a building, the epigenetic code, and the building itself, the synaptic structure. The building may weather, individual bricks may crumble and be replaced with new ones. But as long as the blueprint is preserved, the building can be restored. Memory is durable not because its substrate is unchanging, proteins are constantly being renewed, but because the instructions for restoring the substrate are stored on a more stable medium, in chemical marks on the chromatin. Brady adds one more detail that underscores the depth of this molecular mechanism. DNA in a neuron can temporarily change its conformation during learning. The right-handed helix, the famous Watson and Crick double helix, can locally shift into a left-handed form, so-called zDNA. This conformation is more labile, more mobile, and it makes the DNA more accessible to modifications. Memory, at the very deepest level, changes the very shape of the DNA, not its sequence, but its three-dimensional structure. Brady emphasizes, we have a four-letter genetic code, A, T, G, C. A computer has a binary code of ones and zeros. Already at the level of the code, we have twice as many symbols. And if you consider that each of the four bases can be chemically modified in 4 or 5 different ways, methylation, hydroxymethylation, and others, the combinatorial capacity of the system becomes colossal. We are only beginning to understand how much information can be written in the epigenetic state of a single neuron. And then, in October of 2025, something happened that many neuroscientists consider the most precise demonstration of the molecular mechanism of memory ever achieved. A research group led by Professor Johannes Batt at the Laboratory of Neuroepigenetics at EPFL, the Swiss Federal Institute of Technology in Lausanne, conducted an experiment that brought everything we have been discussing to a single point. David Coder, Lisa Watt, Lillian Glauser and their colleagues used the CRISPR gene editing tool, but not to change the DNA sequence. Instead, they used it to change the epigenetic state of a specific gene in specific neurons. They developed molecular tools that could either silence expression of the ARC gene at its promoter by adding repressive epigenetic marks, closing the chromatin, or, conversely, boost its expression by opening the chromatin and making the gene more accessible. And here is the crucial element. They applied these tools not to all the neurons in the brain, but only to engram neurons. Only those specific neurons that had been active during learning and that stored a specific memory.
To do this, they used a tagging system based on the CFOS gene, an immediate early gene that switches on in neurons when they fire. Only those neurons that were on during learning received the tag, and only in those neurons did the CRISPR tool modify the epigenetic state of ARC. The results were stunning in their clarity. When ARC was epigenetically silenced in the engram neurons, the mice stopped displaying the learned response. They showed no fear when placed in a context associated with an unpleasant memory, as though the memory had been erased. When ARC was epigenetically activated, the memory became stronger. The mice displayed a more pronounced fear response. These effects were observed for both fresh, still labile memories and for fully consolidated memories weeks after learning. And most importantly, the epigenetic editing was reversible. Using a molecular reset mechanism, the researchers could restore the original epigenetic state of the ARC promoter and return memory expression to its initial level. The memory had not been destroyed. It had been muted by an epigenetic switch, and that switch could be flipped back. One gene, one cell type, reversible control of a specific memory. Think about the path we have traveled. Hebb, in 1949, hypothesized that joint activity of neurons strengthens the connection between them. Kandel, in the 1960s and 70s, demonstrated this at a single synapse in a slug. Bliss and Lomo in 1973 discovered LTP in the hippocampus. Over the following decades, scientists deciphered the molecular cascade. NMDA receptor, calcium, CAMK2, AMPA receptors, CREB, ARC, structural changes in dendritic spines. And now, in 2025, Coda and Bat showed that this entire story can be reduced to a single point, the promoter of one gene in one neuron.
Open the chromatin, the memory switches on. Close it, it switches off. Open it again, and it comes back. We have descended from the feeling you experience when you remember the smell of tangerines, through the map of the brain, through synaptic connections, through molecular cascades, down to a single methylated stretch of DNA in the nucleus of a single neuron in your hippocampus. The smallest nesting doll is in our hands, and it turned out not to be empty. Inside it is a molecular toggle that can be flipped, but this smallest doll opens a door to a question that reaches far beyond neurobiology. If memory is written in epigenetic marks, and epigenetic marks can be copied during cell division, does that mean memory can be passed from cell to cell? And if it can, then can it be passed from generation to generation? The Dutch hunger winter. The winter of 1944 to 1945. The final months of World War II in Europe. The German occupation of the Netherlands.
Blockade. Famine. The daily ration for the population in the western provinces dropped to about 400 calories a day, roughly 2 slices of bread and an onion. About 20,000 people died of starvation. Those who survived spent the rest of their lives with the consequences of the famine: elevated risk of diabetes, cardiovascular disease, psychiatric disorders. But here is what stunned researchers when, decades later, they began studying this tragedy through the lens of epigenetics. The consequences of the famine were not limited to those who lived through it. The children of women who were pregnant during the hunger winter also showed elevated rates of metabolic disorders. And their grandchildren did too. People who had never gone hungry, who were born in the prosperous decades after the war, carried in their cells the molecular trace of a hunger they themselves had never experienced. When scientists compared the epigenetic state of the genomes of those conceived during the famine with a control group, they found a difference. Specifically, altered DNA methylation on the IGF2 gene, a gene regulating fetal growth. The methyl marks on this gene were different in people whose mothers had starved during pregnancy, and different even in the next generation. The molecular signature of famine was legible in the epigenome of people who had not yet been born when the hunger ended. The memory of famine had been written in the epigenetic code and transmitted across generations. In parallel, in the laboratory, scientists obtained more direct evidence of the same phenomenon. Brian Diaz and Kerry Bat Ressler at Emory University conducted a series of experiments on mice that became one of the most discussed publications in the field of epigenetics in the last decade. They trained male mice to fear a specific odor, acetophenone, which smells like cherry blossoms. The training was classical. The odor was paired with a mild electric shock. The mice quickly learned to fear the smell and displayed a fear response every time they detected it. Then the researchers studied the offspring of those mice, offspring that had never undergone training, that had never received a shock, that had, in principle, no reason whatsoever to fear the smell of acetophenone. These mice also displayed heightened sensitivity and fear to that odor, and their offspring, the second generation, did too. The mechanism was this. The trained fathers showed changes in DNA methylation on the OLFR151 gene, which encodes the olfactory receptor that recognizes acetophenone. These changes were present in their sperm. In the offspring, the same methylation pattern was replicated. And, this is especially telling, in their olfactory epithelium, the number of neurons expressing that particular receptor, OLFR151, was increased.
The noses of these mice were literally rebuilt for detecting the odor their fathers had feared. Experience changed the epigenetic state of germline cells, the sperm. The epigenetic state was transmitted to the offspring, and this epigenetic inheritance physically altered the nervous system of the descendants, making them more sensitive to a stimulus that had been dangerous for their ancestors. Now recall the plants we mentioned. Trees that survived drought accumulate epigenetic marks on the promoters of stress response genes, specifically H3K4-ME3, which makes the chromatin more accessible. When the next drought arrives, those genes switch on faster. The tree responds more quickly and more effectively. And here is what is striking. In poplars, this drought memory is transmitted through the division of cambium cells, the tissue that produces new wood and bark each year. The cambium cells that exist this year have personally never experienced last year's drought.
The cells that live through that drought have long since stopped dividing and become part of the wood. But the epigenetic memory was copied during cell division and passed to daughter cells and to their daughter cells, and so on. The tree remembers the drought through cells that were not present for it. Before we leave the chapter on inheritance, let us step back and look at the full picture, because what we see changes the very definition of memory. Every cell in your body remembers what type of cell it is. Your body contains roughly 37 trillion cells, and virtually every one carries the same copy of your genome. A liver cell and a neuron have an identical genome, but they look completely different, work completely differently, and express completely different sets of genes. How does a liver cell know it is a liver cell and not a neuron?
It remembers. It remembers through epigenetic marks, the pattern of DNA methylation and histone modifications that determines which genes are accessible and which are locked. That pattern was established during embryonic development when a precursor cell decided to become a liver cell, and since then, every division has copied that pattern along with the DNA. Daughter cells inherited not just the genome, but its interpretation. This is cellular memory in the most precise biochemical sense. Your immune system is another astonishing example of memory without neurons. When your body first encounters a pathogen, a bacterium, a virus, a parasite, the immune system mounts an adaptive immune response. It generates lymphocytes with receptors specifically matched to the antigens of that pathogen, multiplies them, and destroys the infection. This process takes days or weeks.
But after the infection is defeated, some of the activated lymphocytes survive as memory cells, long-lived cells carrying a molecular record of the encounter in the structure of their receptors and in their epigenetic state. If the same pathogen shows up again, the memory cells recognize it immediately and mount a response within hours, not weeks. The response will be faster, stronger, more precise. This is immunological memory, and it is the principle behind vaccination. By introducing a safe version of the pathogen, we form immunological memory without the cost of actual infection. Note: these cells have no axons, no dendrites, no synapses. There is nothing that a neurobiologist would call a memory system. And yet, they store information about past encounters with threats and use that information for a more effective response in the future. The same logic as in a hippocampal neuron
but implemented by entirely different means, or rather, by the same epigenetic means, just in different cells. The same molecular language of memory, methylation, acetylation, chromatin remodeling, is universal. It is used by a tree remembering drought, a lymphocyte remembering a virus, and a neuron remembering the smell of tangerines. The differences are in scale, in complexity, in the number of components involved. But the principle is one. Change the accessibility of genes through chromatin modification so that the cell responds differently in the future than it did before the experience. To remember, in the language of molecules, means to alter the chromatin configuration of a cell so that its future behavior differs from its past. And this fundamental principle works from bacterium to human. We have reached the very bottom of our nesting doll.
We have descended from the feeling of smelling tangerines through the map of the brain, through synapses, through molecular cascades, through the epigenetic code, down to a mark on the DNA that can outlast generations. Now let us begin the journey back up, from molecule to thought, because we still have not answered one of the most mysterious questions. What exactly happens when you remember? Not when you learn, but when you remember. When an already recorded memory somehow rises back up from the depths of chromatin, from the architecture of synapses, from the hippocampal map, into your consciousness. How does a skeleton spread across thousands of shelves reassemble itself into a living creature? And why does each reassembly produce a slightly different creature than the last? Think again of the childhood smell we started with. Tangerines, your grandmother's kitchen, the light from the window, the warm feeling. You just summoned that memory again.
But was it exactly the same as 15 minutes ago?
When you recalled it at the beginning of our conversation? Most likely not. Maybe this time a new detail emerged, or maybe something was lost. Maybe the emotional tone shifted slightly, became softer, or a little sadder, because between the first and second recall, you spent time thinking about how the brain works, and that colored your return to the childhood scent. And here is the most astonishing part of this story. Your memory truly did change. Not just subjectively, not just it seemed that way. Physically, at the level of neurons and synapses, every act of remembering is an act of rewriting. Let us break down how this works. When you remember, the hippocampus initiates pattern completion. A small fragment, a smell, a sound, an image, an emotion, activates hippocampal neurons linked to that specific memory.
Those neurons send signals back to the cortex, activating fragments of the memory scattered across different zones. The visual fragment in the occipital cortex, the auditory one in the temporal cortex, the olfactory one in the olfactory cortex, the emotional one in the amygdala. Gradually, from the activation of 20% of the neurons that were active during the original experience, a picture is reconstituted, pale, incomplete, but sufficient for your consciousness to recognize it as a memory. But here is what happens in parallel with this reconstitution. When a memory is retrieved, when the engram neurons are reactivated, it enters a labile state. It becomes vulnerable. For a brief time, while the memory is unfolded in active memory, it can be modified. New information available at that moment, your current mood, your present context, other thoughts running through your head, can be integrated into the memory.
And when the memory is folded back up and returned to long-term storage, it goes through a process neuroscientists call reconsolidation. Reconsolidation is reconsolidation. The memory is stabilized again, but now accounting for the changes introduced during retrieval. Protein synthesis is needed again. Synaptic changes are needed again. Epigenetic modifications are needed again. In essence, every time you remember, you partially disassemble the memory and reassemble it. And what is reassembled is no longer quite what was taken apart. It is an updated version, incorporating elements of your present. A study published in the journal Neurons in February of 2025 by the group of Lei Kan Yu Jun He and colleagues at Tsinghua University showed that during reconsolidation, something even more radical occurs than simple updating. When a mouse recalled a fully consolidated, long-established memory, one that had already been transferred from the hippocampus to the cortex and no longer depended on the hippocampus for its storage, the hippocampus became involved again. But not through the same set of neurons that had originally encoded the memory. Through a new set. A new group of neurons was recruited specifically for the reconsolidation process, for updating the old memory with new context. And here is what was critical. These new engram neurons arose through neurogenesis, the birth of new neurons from stem cells in the hippocampus. When the researchers suppressed neurogenesis, the old hippocampal engram did not deactivate, the new one was not recruited, and memory updating did not occur. The brain could not simply overwrite the past. It needed new cells to represent the past in a new context. Every time you remember something, your brain may be creating new neurons to update that memory.
The past you store in your hippocampus is not a fixed record. It is a continuously updated reconstruction, built at the intersection of what happened and what you know now. That is why Brian Greene's memory of the stroller and the hot dog became a third-person view. Over decades of retelling and remembering, his brain reconsolidated that episode again and again, weaving in new elements, including the perspective of a narrator who watches himself from above. That is why eyewitness testimony is so unreliable. Every time a witness retells what they saw, their memory passes through reconsolidation and can be altered by the investigator's questions, the atmosphere of the interview, even the expression on the interviewer's face.
And that is why PTSD therapy through reconsolidation has shown such promise. If a patient summons a traumatic memory in a controlled, safe environment, and at that moment pharmacologically dampens the emotional component, for example with propranolol, a beta blocker that reduces adrenergic activation, then during reconsolidation, the memory can be reassembled with a lower emotional load. The memory of the event remains, but the sharpness of the pain diminishes. You remember what happened, but it no longer tears you apart from the inside. Gail Robinson, the neuropsychologist, formulates it carefully. There is a level of reconstruction, a level of reconfiguration. Every time you pull a memory out of the drawer, you shuffle it a little. Your thoughts, your state at that moment, all of that seeps in and mixes with what was there. So next time, on the beach, if you recall this evening, the beach will weave itself into the memory too, and they all feel equally real.
Oliver Baumann adds, maybe that is what real actually is. A memory is real for you as an individual, even if it does not perfectly correspond to objective events. It is your version of the past, and it shapes your identity no less effectively than a perfectly accurate recording that never existed. But do not despair over this. Robinson notes that the core of a memory, its basic scaffold, is usually quite stable. The peripheral details change, the color of the walls, the exact words, the sequence of small events. But the essence, what happened, who was there, and what emotion it produced, generally holds. And that is enough for memories to perform their primary function, guiding future behavior. Because memory, as several participants at the Brisbane panel aptly noted, is not an archive. It is a bridge from past to future. We do not remember in order to be nostalgic. We remember in order to act. Your brain preserves the past because the past is the best predictor of the future. Robinson draws an important practical conclusion that has to do with aging. As we get older, we tend to shift to autopilot. Habits replace conscious decisions. Familiar routes require no attention. Routine swallows up novelty. This is efficient. The brain saves resources by using accumulated experience to automate everyday tasks. But there is a cost to that efficiency. Automatic behavior does not create new episodic memories. If every day is like the one before, none of them stands out from the stream, and none receives enough emotional tagging for robust encoding. Robinson and Bauman both emphasize, if you want to keep memory active, the key is mindfulness and novelty. New experience, conscious attention, emotional engagement, all of these send the brain the signal, this moment is worth encoding. Bauman adds, over time our brain increasingly relies on prior experience to fill in the gaps in current perception.
New information is absorbed faster when it sits on a foundation of prior experience. But we pay for this with reduced attention to details that seem familiar. This is a normal, evolutionarily justified process. But it explains why years in adulthood feel subjectively shorter than years in childhood. In childhood, every day brought something new. Every event was tagged with emotion and attention, and each was encoded as a separate episode. In adulthood, there is less novelty, more automaticity, and days blur together in memory. But this can be changed. Not with gene therapy, not with implants, but with a simple, conscious decision. Pay attention. Be here. Notice what is happening. Because your brain records only what you pay attention to and what you feel. Now let us turn to the other side of this process. We have talked a great deal about remembering, about how experience becomes memory, how memory is strengthened by emotion, how it is written in synapses and chromatin. But we have said almost nothing about forgetting. And forgetting, as it turns out, is every bit as interesting and every bit as important as remembering. Borges wrote a story called Funes the Memorious, about a man who remembered absolutely everything. Every leaf on every tree he had ever seen, every cloud in the sky, every instant of every day. And it turned out to be a curse, not a gift. Funes could not generalize. He could not abstract. Every dog he saw was unique to him. He could not group them into the category dogs, because he remembered the specific appearance of every specific dog. He was buried under detail, crushed by the precision of his own memory, and he could not sleep. A real-world parallel of Funes exists. Gail Robinson works with people who have what is called HSAM, highly superior autobiographical memory. There are roughly 60 to 100 such people in the world. They remember every day of their lives. You can name any date and they will tell you what they did that day, what the weather was, what they ate, whom they talked to. Not major events, ordinary days, Tuesdays and Thursdays that you and I forget as soon as they end. Robinson worked with one such woman in Australia, the only person in the country with confirmed HSAM. Her memories surface constantly, intrusively, uninvited.
She sleeps poorly because the stream of memories is hard to shut off. When Robinson showed her a black and white photograph of a beach, a standard clinical test in which the patient is asked to describe the image, this woman began describing colors, sounds, smells. On a black and white photograph, she was automatically filling in the image from her own experience, saturating it with sensory information that was not in the photograph. Robinson notes something unexpected. On neuroimaging, this woman's brain does not differ from the brains of ordinary people. The hippocampus is not enlarged.
The amygdala is standard size. There is no super memory organ. The difference is probably in something subtler, perhaps in how the forgetting system works, or, more precisely, in the fact that it does not work as it should. Because forgetting, as research in recent years has shown, is not passive decay, not a fading signal, not a bleaching of the record. Forgetting is an active, regulated, purposeful process. The brain spends energy and molecular resources on erasing memories, and this makes deep evolutionary sense. A memory system that preserved everything with equal precision would be functionally paralyzed. It would be buried in noise, millions of insignificant details that would get in the way of quickly finding important information. Forgetting is not a defect of the system. It is a filter that sifts out noise and preserves signal. The brain forgets what you are unlikely to need, to free resources for what you will.
Hermann Ebbinghaus, the German psychologist, ran a famous experiment back in 1885, memorizing nonsense syllables, three-letter sequences without any meaning, and testing how many he remembered after various intervals. His forgetting curve showed that we lose roughly 50% of new information in the first hour, about another 20% by the end of the day, and then forgetting slows, reaching a plateau. After a week, about 25% of what was originally learned remains. Of course, nonsense syllables are an extreme case. They lack both emotion and meaning and context, all the factors that enhance consolidation. Meaningful, emotionally charged information is forgotten much more slowly. But even accounting for that, the volume of information our brains process daily and release daily is staggering. And this is an evolutionarily sound strategy.
Bauman gives a simple and convincing example. If you park your car in the same lot every day, but in a different spot each time, your brain faces a dilemma. Storing information about every single day's parking is wasteful. 10,000 memories of exactly where the car sat only interfere with quickly finding today's location. The brain solves this problem elegantly. It keeps the freshest record and allows older records to interfere with each other and fade away. Bauman emphasizes, forgetting is not a malfunction. Forgetting is a feature. We forget in order to function. We forget in order to generalize. We forget in order to extract patterns from the noise of specific instances. You do not remember every dog you have ever seen, but you know what a dog generally looks like. That generalization is possible precisely because the details of specific encounters were discarded and the common features preserved. Forgetting details is the precondition for generalization.
And generalization is what makes your knowledge of the world useful, not just vast. Borges understood this intuitively. His Funes, who remembered everything, could not think. To think is to forget differences, to generalize, to abstract, Borges writes. Funes could not generalize because he could not forget. His memory was perfect, and that is precisely why it was useless. Tim Brady of the Queensland Brain Institute describes one of the mechanisms through which the brain maintains and suppresses memories, and it involves the neurotransmitter GABA, gamma-aminobutyric acid, the main inhibitory neurotransmitter in the brain. Brady proposes that memories are, in a sense, always
online, always in a state of potential activation, but actively suppressed by inhibitory signals. GABA acts as a brake. It prevents activation of the engram neurons, keeping the memory below the threshold of consciousness. When the inhibitory signal weakens, through an external stimulus like a smell or a melody, or through an internal process like a conscious effort to remember, the engram neurons fire and the memory rises into awareness. If this model is correct, HSAM may be linked not to supermemory, but to weakened inhibition. Not to these people remembering better, but to their forgetting worse. The brakes are not working as they should, and memories surface uninvited. And organisms far from us, nothing like humans, also have active forgetting systems. Caenorhabditis elegans, a tiny roundworm about a 25th of an inch long with just 302 neurons, can learn, associating odors with the presence or absence of food, habituating to repeated stimuli. And, as a study published in Nature Neuroscience in December of 2025 showed, it also actively forgets. Researchers found that worms kept at low temperatures or treated with lithium forgot more slowly. The worms remembered learned odor associations significantly longer than usual. The mechanism involved the diacylglycerol signaling pathway in olfactory neurons, a specific molecular cascade that normally erases memories on a set schedule and that can be blocked. Forgetting in C. elegans is not passive degradation of a trace. It is an active program that runs on a schedule and can be slowed or accelerated by specific molecular interventions. And if forgetting is actively regulated, even in an organism with 302 neurons, it is hard to imagine that it is not regulated just as precisely, or even more elaborately, in a brain with 86 billion. There is one more mechanism that links forgetting to sleep and that we cannot pass over in silence, because you may be approaching the state in which this mechanism is about to kick in. In 2012, a team of scientists, led by Mike and Nedergaard at the University of Rochester, described something she called the glymphatic system. During sleep, especially during deep, slow-wave sleep, the spaces between neurons in the brain expand by about 60%. Cerebrospinal fluid begins actively flushing through these widened channels, washing away metabolic waste accumulated during the day. Among that waste is beta amyloid, a protein that, when it accumulates, forms the plaques characteristic of Alzheimer's disease. The brain literally washes itself during sleep. And if you are chronically sleep-deprived, that laundry does not happen fully, and neurotoxic waste accumulates. The link between chronic sleep deprivation and elevated risk of neurodegenerative diseases may be partially explained by exactly this. Sleep is not a luxury and not wasted time. Sleep is maintenance of the memory system.
During slow-wave sleep, the hippocampus replays the day's events, repeating patterns of neural activity recorded during waking, but in accelerated mode. It is as though the brain is doing a quick summary of the day, selecting what to transfer to long-term storage. Studies in rats demonstrated this with striking directness. When a rat explores a maze during the day, specific neurons in its hippocampus fire in a specific sequence, each neuron coding a specific location in the maze. At night, during slow-wave sleep, the same neurons fire in the same sequence but faster, like a fast-forward replay. The hippocampus literally runs through the day's route, reinforcing it. Simultaneously, the hippocampus sends signals to the cortex, gradually handing off responsibility for storing those memories. Over time, over weeks and months, The memory becomes less dependent on the hippocampus and more dependent on the cortex.
That is why Henry Mollison remembered his distant past, but could not form new memories. His old memories had already been consolidated in the cortex and no longer needed the hippocampus, while new memories could not get through the hippocampus for initial processing. Sleep also performs another important function beyond consolidation. During REM sleep, the stage where we dream, the brain appears to test consolidated memories, integrating them into existing knowledge networks and looking for connections with information already known. Dreams, for all their apparent senselessness, may be a byproduct of this integration process. The brain combines fragments of new and old experience into bizarre collages. And those collages are what we see as dreams. Veronica O'Carroll, with her 3 decades of experience in psychiatry and neuropsychology, adds yet another dimension. She stresses that memory is not only a brain phenomenon, it is experienced in the body. The emotions that accompany memories have physical manifestations: a racing heart, tightness in the chest, warmth, chill, goosebumps. And these bodily sensations are not simply extras added to a memory. They are an integral part of it. Memory and body are linked in a loop, and sometimes it is the bodily sensation that becomes the key triggering the memory rather than the other way around. You smell something and a picture surfaces. You hear a melody and a feeling returns. Body and brain work together, and their partnership runs deeper than we usually realize. Now let us connect all the levels. We have taken the nesting doll completely apart, from the experience of a memory, through brain structures, through synapses, through molecules, through the epigenetic code, through transgenerational transmission, and back up again, through reconsolidation and forgetting. And one last thing remains to tell: the story of those who physically found a memory, found a specific group of neurons that stores a single, specific episode of your life. This group is called the engram, and the story of its search is a century-long detective case with a stunning denouement. The term engram was proposed in 1904 by the German biologist Richard Semon. Semon defined the engram as the physical trace left in nervous tissue by experience, which, when reactivated, reproduces the memory of that experience. Put simply, a memory is a concrete thing living in a concrete place in the brain. It is material, and in principle, it can be found. Semon's work was largely ignored during his lifetime, but his idea was taken up by the American psychologist Carl Lashley, who devoted 3 decades, from the 1920s through the 1950s, to a systematic search for the engram in the rat brain. His method was straightforward. Train a rat on a task, say running a maze, then surgically remove a section of cortex and check whether the memory survived. If yes, remove another section, and another, until the memory disappeared, at which point the removed section would be the location of the engram. Lashley never found the engram. He removed more and more brain tissue, and performance worsened. But the memory never vanished completely with the removal of any single region. The more tissue he removed, the worse the performance. But the decline depended on the volume of tissue removed, not on which particular region was taken out. Lashley arrived at his famous and exasperated conclusion that learning is just not possible at all, meaning that if memory is uniformly distributed across the cortex and localized nowhere, its very existence as a physical trace is problematic. Lashley was wrong, but wrong for an understandable reason. He could not have known what we know now. The engram really is distributed across many neurons, and the contribution of each individual neuron is so small that it cannot be detected by the crude method of removal. By cutting out one area of cortex, Lashley was removing only a portion of the engram, and the remainder was sufficient to sustain the memory, albeit in weakened form. To find the engram, tools were needed that did not exist in Lashley's lifetime. Tools that could not merely remove tissue, but could tag the specific neurons active at the moment of learning, and then selectively activate or suppress only those neurons. Such tools arrived in the early 21st century. They are called optogenetics, a technology for controlling neuron activity with light. The idea: you genetically modify certain neurons so they express a light-sensitive protein, channel rhodopsin. This protein is an ion channel that opens when illuminated with blue light. When you aim a beam of light at a modified neuron, the channel opens, ions rush in, and the neuron fires. Light turns on the neuron. In 2012, Susumu Tonegawa and his colleagues at the Massachusetts Institute of Technology used this technology to do what Lashley could only have dreamed of. They genetically engineered mice so that neurons active during a specific learning event, contextual fear conditioning, automatically expressed channel rhodopsin. The marker of activity was the C-FOS gene. When a neuron fired during learning, C-FOS switched on, and along with it, channel rhodopsin expression was triggered. Only those neurons that participated in recording that specific memory received the light-sensitive channel. Then, days later, the researchers shone blue light into the mouse's hippocampus through a thin fiber optic cable. The light activated only the engram neurons, and only them. There was no other stimulation, no context,
No smell, sound, or place that the mouse could associate with the frightening experience. And the mouse froze. It displayed the classic fear response, freezing, in a neutral environment where it had nothing to fear. It remembered an experience that was not happening. Activation of the engram neurons, and only them, was sufficient to reproduce the memory. The engram was real. It was physically embodied in a specific population of neurons. It could be found, tagged, and turned on. In subsequent years, the architecture of the engram was studied in greater detail, and the picture turned out to be more complex and more elegant than a simple club of neurons that talk only to each other. A study published in the journal Science in 2025 by the group of Oyito and colleagues used three-dimensional electron microscopy for nanometer-scale reconstruction of synaptic connections between engram neurons in the CA3 and CA1 zones of the mouse hippocampus.
And the result was unexpected. Intuitively, you would expect engram neurons to form a tightly connected group, a clique, where each member talks mainly to other members of the engram. Neurons that fire together wire together, right? So the neurons of a single memory should be tightly bound to each other, but Oyito and colleagues found something different. Engram neurons in CA3 did not show a strong preference for forming connections, specifically with engram neurons in CA1. Instead, engram neurons expanded their broadcast radius. They increased the number of what are called multi-input boutons, presynaptic terminals that simultaneously contact several postsynaptic cells. An engram neuron did not create a closed circuit with other engram neurons. It expanded its connectivity, contacting a larger number of postsynaptic partners, increasing its influence in the network. The engram is not a closed club. The engram is a broadcasting station.
It does not store the memory inside itself, like a file on a disk.
It launches a pattern of spreading activation that, radiating through hippocampal and cortical networks, recreates the full multidimensional experience: visual, auditory, tactile, olfactory, emotional. The memory you consciously experience is the result of this spreading wave, not the engram itself. The engram is the starting point, the detonator that triggers a chain reaction of restoration. That is why Lashley could not find the engram by cutting out pieces of cortex. He was removing fragments of a distributed picture, but the detonator in the hippocampus remained intact, and the remaining fragments were enough for an incomplete but recognizable picture to still come through. And now we can assemble the entire structure, from molecule to thought. 1 second of remembering, and every level we have examined works simultaneously, nested one inside the other, like those very nesting dolls. You smell tangerines. An olfactory receptor on the lining of your nose catches a molecule, sends a signal to the olfactory cortex. The olfactory cortex activates neurons associated with that smell. The hippocampus, receiving the signal, initiates pattern completion. It searches for the index card linking that smell to a specific episode. It finds one. It sends signals back to the cortex. Activate the visual fragment, this particular light from the window. Activate the auditory fragment, these particular sounds. Activate the tactile fragment, this particular sensation. The amygdala adds emotional coloring, warmth, tenderness, perhaps a touch of sadness. Engram neurons expand their broadcast radius through multi-input boutons, and a wave of activation radiates through cortical networks, restoring roughly 20% of the original experience, enough for your consciousness to recognize it. At the level of synapses, AMPA receptors on potentiated connections admit more sodium. The signal on those connections is louder. That is why those particular neurons fire first, why that particular picture reconstitutes, and not a million other possible pictures. At the level of molecules, CAMK2 phosphorylates proteins, reinforcing the activated connections. NMDA receptors, if the activation is strong enough, admit calcium, triggering the next wave of molecular events. At the level of the nucleus, the ARC gene, whose promoter carries epigenetic marks from previous activations of this memory, switches on quickly. Non-coding RNAs begin their journey from nucleus to synapse, and simultaneously, reconsolidation. The memory, activated but already slightly altered by your current state, your thoughts today, your mood right now, is being rewritten. It will be a little different the next time you recall it. The tree has grown one more branch. All of this in less than a second. A cascade from cortex to chromatin and back. We have come a long way from the smell of tangerines to methyl groups on DNA. But the story of memory does not end with understanding. It is approaching a point where understanding begins to turn into the possibility of intervention. And that possibility raises questions that 10 years ago were science fiction, and today stand on the threshold of clinical reality. Michael Kahana, professor of psychology at the University of Pennsylvania, leads the computational memory lab and works on something that sounds like a plot from the future: memory prosthetics. His approach is based on an idea we have discussed. Memory fluctuates. Within a single day, within a single hour, your ability to encode and retrieve rises and falls. These are not random fluctuations. They reflect changes in the brain's electrical activity that can be measured through implanted electrodes.
Kahana and his team developed a system that does two things. It decodes, meaning it reads the brain's electrical signals and predicts when memory will be good and when it will be poor. And then, in the moments when memory is predictably weak, the system delivers gentle electrical stimulation to certain brain regions, nudging neural activity toward the pattern that corresponds to good memory. The patient does not feel the stimulation. They do not know when it is being delivered. But memory test scores improve, on average, by about 19%. Kahana explains that 19% is roughly half the memory impairment caused by a moderate to severe traumatic brain injury. For a third of patients, the effect is even stronger, nearly full restoration of lost function. For another third, there is no effect. For the remaining third, the result is intermediate. Kahana envisions a more distant horizon. If such devices become miniaturized enough, we will have a system that does not merely treat memory impairments, but generates an enormous database on how the brain encodes and retrieves information in the real conditions of life, not the sterile setting of a laboratory. Patients with implanted electrodes will walk the streets, talk with friends, watch movies, argue, fall in love. And all the while, the device will be recording the electrical activity of their brains and correlating it with results from periodic memory tests. The data, encrypted and transmitted to the cloud, will create what Kahana compares to a Hubble telescope for memory neuroscience, an instrument that would allow testing theories on scales inaccessible to traditional laboratory work. Edward Chang, for his part, has already demonstrated that the neural code of speech can be decoded in real time, translating the brain's electrical activity back into the words a person intends to say. His Bravo clinical trial involved a young man who, after a car accident, lost the ability to speak due to damage to the brainstem. His intention to speak was preserved in the cortex, but it could not reach the muscles of the vocal tract. Chang's team learned to read those intentions from the cortical surface and translate them into synthesized speech. The words sounded fuzzy, blurred, but they were discernible. A man who could not utter a single word spoke again, through a machine reading his brain. If speech decoding is already possible, memory decoding is the next logical step. And if the CRISPR tools of Coda and colleagues allow pinpoint epigenetic editing in specific neurons, and Kahana's devices allow precise reading and stimulation in real time, then the prospect before us is both breathtaking and a little unsettling. Here is what stands on the horizon. Technology capable of reading, writing, and editing memories. Not all memory, not through crude stimulation of the entire brain, but precisely, selectively, at the level of specific neurons, specific genes, specific epigenetic marks. Weaken a traumatic memory, strengthen a fading one, update an outdated one, restore a lost one. It sounds impossible, but every element of this technology has already been demonstrated in the laboratory, in animals. Bridging the distance from laboratory to clinic is a question of engineering, safety, and ethics, not of fundamental possibility. On another front, work with the epigenetics of memory opens prospects that are even more radical. A study published in the journal Neurons in February of 2026 showed that partial epigenetic reprogramming of engram neurons using Yamanaka factors, the very proteins that allow a differentiated cell to be returned to a stem cell state, can restore memory in old mice to the level of young ones. The reprogramming was targeted specifically to those neurons that had been active during learning, and only to them. And it restored both the cellular properties of those neurons and the behavioral expression of the memories they stored. The aging of memory, at least in part, turned out to be an epigenetic problem.
The chromatin of aging neurons gradually changes configuration. Genes that should be accessible become less so. The molecular machinery of synaptic plasticity works with reduced efficiency. But if you return the chromatin of these specific neurons to a younger state, not the entire brain, just the memory neurons, function is restored. And the CRISPR tools that Coda and colleagues demonstrated in 2025, in principle, allow not only silencing and amplifying memories, but selectively editing them. Imagine a therapy for PTSD in which, instead of propranolol suppressing the emotional component during reconsolidation, a molecular tool is used that specifically weakens the epigenetic mark on the ARC promoter in precisely those neurons that store the traumatic memory. The memory is not destroyed, it is muted. The sharpness of the pain is reduced, the context is preserved, but the destructive force of the traumatic memory is diminished. And it is ethics that poses the hardest questions. If your memories are you, and we have seen that this claim has serious grounds, then altering a memory is altering you. Who decides which memories can be erased? Who determines which should be strengthened? Does a person have the right to forget what they themselves want to forget? And if their desire to forget is itself a product of the same traumatic experience they want to erase, how free is that choice? Michael Halassa from MIT brings yet another perspective to the conversation about memory, one that connects everything we have discussed to a broader question about why brains exist at all. One of the main goals of having a brain, says Halassa, is to plan the next action. Without the ability to move and act in the world, there is no point in having a brain. That is why plants do not have brains. And over the course of evolution, organisms that better planned their sequences of actions developed larger brains, more complex internal models of the world. And memory is what connects past action to future planning. O'Carroll answers the question of identity with the perspective of a clinician who has spent 30 years observing how damage to memory changes people. Memory binds us, she says. We live in a moment of consciousness.
Our memory and past exist in the present. And in that conscious instant of the present, we predict the future. Memory is what connects all three, past, present, and future, in one experience we call the self. She adds something that goes beyond individual psychology. The human race is very much like memory. We are living in our current situation. We are well aware of the past, and we are trying to prevent past mistakes in our future behavior. I think we evolve as a species in exactly the same way as we evolve and develop personally. Memory binds us together. Halassa suggests thinking about the brain in terms borrowed from David Marr, on 3 levels of description. The computational level. What task is a given neural process solving? The algorithmic level. What steps does the brain take to solve that task? And the implementation level. How are those steps carried out in actual neurons? For memory, the computational task is storing and retrieving information about the past to guide future behavior. The algorithm is Hebbian plasticity, LTP, pattern completion, consolidation. And the implementation is NMDA receptors, calcium, CAMK2, CREBARC, epigenetic modifications. We have walked through all 3 levels tonight. And the beauty of the picture, as Halassa notes, is that the 3 levels are beginning to converge. We are beginning to understand how the algorithms of the mind are implemented in the physical substrate of neurons. But the understanding is far from complete.
We may be missing something fundamental in our description of science in general to answer questions about consciousness, Halaser admits. And there is a collective dimension to memory that deserves mention. Think of the events of September 11th, 2001.
Everyone who was alive and old enough to understand that day remembers where they were and what they were doing when they heard the news. These are personal, individual, unique memories for each person. And at the same time, they are collective, shared, unifying. We all watch the same footage again and again, and every repetition strengthened consolidation. Those images became part of a collective memory, and because of them, strangers who have never met share a common experience. Memory, says O'Carroll, is ultimately about connection. Connection between neurons. Connection between the moments of your life. Connection between you and other people. Connection between generations through the very epigenetic marks that carry experience from parents to children. And connection between species through the molecular toolkit that evolution conserved from the sea slug to the human being. One of the most important lessons the history of memory research teaches is humility. Every generation of scientists was confident they had arrived at the final answer, and every generation discovered the problem runs deeper than they thought. Lashley was certain the engram could be found by cutting the right piece of cortex. He searched for 30 years and never found it. Penfield was sure memories were recorded on the cortex, like grooves on a record. He was closer to the truth, but still oversimplifying. Hebb proposed a beautiful principle, but had no tools to test it. Candell found the molecular mechanisms in a sea slug, and skeptics doubted for decades that those mechanisms applied to vertebrates. Tonegawa found the engram, and it turned out to be organized differently than anyone expected. Not a closed club, but a broadcasting station. Coda and colleagues showed you could switch a memory on and off with a single molecular toggle, and instantly new questions opened up that we do not yet have the answers to. Every answer spawns new questions. Every closed door leads to a room with 3 new doors. And that is perhaps the most beautiful thing about the science of memory. The more we learn, the clearer it becomes how much we still do not know. We have descended to the level of a single methylated cytosine on the promoter of a single gene. And even here, at the very bottom of our nesting doll, questions remain. We stand on the threshold of an era in which memory, this most intimate, most personal of all biological processes, becomes accessible to technological intervention. We can read the brain's electrical signals and predict when memory will be good and when it will be poor. We can stimulate the brain and improve memory by 19%. We can switch specific memories on and off through epigenetic editing of a single gene. We can rejuvenate aging memory neurons with Yamanaka factors. Each of these capabilities opens doors, and each raises questions we have not yet learned to ask. But these questions, for all their gravity, should not stop us from understanding. Understanding how memory works is the foundation on which responsible decisions about how to use that knowledge can be made. And the path to that understanding, the path we have traveled tonight, is itself proof of what the human mind is capable of when it turns its chief instrument, curiosity, on itself. Now let us return to our big questions and check whether we have answered them. How does your brain decide which moments become memories and which vanish? Through a tagging system, emotion from the amygdala and attention from the prefrontal cortex
determine which episodes get priority during consolidation. Dopamine, serotonin, norepinephrine act as molecular markers of importance that strengthen LTP and facilitate CREB activation. Why do you remember the smell of tangerines but not lunch on Tuesday?
Because the tangerines were tied to an emotionally significant context: childhood, celebration, people you love, and received powerful tagging from the amygdala. Tuesday's lunch passed without emotion and without conscious attention, and the brain rationally chose not to spend resources on its consolidation. Where does a specific memory live, and can you touch it? A memory lives in distributed form. Its fragments are stored in various cortical zones, and the hippocampus holds the index card binding them together. The engram, the group of neurons active during recording, is the detonator that launches the restoration of the full picture. And yes, the engram can be touched. Tonegawa activated specific memories with a beam of light, and Koda and colleagues with a switch on a single gene. What happens at the molecular level when you remember? A cascade from NMDA receptor to chromatin. Calcium, calmodulin, CAMK2. Phosphorylation of AMPA receptors, CREB, ARC. Structural strengthening of the synapse. And at the deep level, epigenetic modifications on the ARC promoter. Methylation, acetylation, non-coding RNAs. Why does the same memory feel different each time? Because every memory, upon retrieval, passes through reconsolidation. It is unfolded, updated with the current context and mood, and refolded, already slightly changed. New neurons, born through neurogenesis, are recruited to represent old experience in a new context. And if your memories are constantly being rewritten, what does that mean about who you are? It means you are not a fixed point. You are a process, a continuous stream of rewrites, updates, consolidations, and forgettings that every night recreates your story based on what was and what you know now. And that process is the very one happening inside you right now, as you lie here and listen. And here you are, at the end of our journey, or nearly the end, because while you lie here, the journey continues inside you. Right now, your brain is doing what we have been talking about all evening. The neurons that were active while you were listening have begun the quiet work of consolidation. Calcium that passed through NMDA receptors at synapses, active during your listening, has triggered the cascade: calmodulin, CAMK2, phosphorylation of AMPA receptors. The connections that were engaged have grown a little stronger. If anything you heard triggered an emotion, surprise, interest, maybe a slight vertigo at the scale of it all, the amygdala tagged those spots. Remember this one firmly. In a few hours, when you sink into slow-wave sleep, your hippocampus will begin replaying the recordings of the day, quietly, inside itself, without your knowledge. It will be sending fragments to the cortex for long-term storage. Some of those fragments may be the image of a sea slug with its giant neurons, maybe the thought of a tree that remembers drought, maybe the fact about CRISPR editing of memory will travel from the hippocampus to the cortex and become part of your semantic knowledge. You will not remember when exactly you learned that the NMDA receptor works as a coincidence detector, but you will know it. The fact will detach from the episode and become part of your picture of the world. Or maybe tomorrow morning you will not remember much of this at all. And that is fine too. Your brain will decide for you what to keep and what to let go. The forgetting system is just as important a part of your cognitive architecture as the remembering system. It sifts out the noise. It frees resources for tomorrow. It makes you capable of living without drowning in detail. But something will probably stay. Maybe an image. Maybe a feeling. Maybe the moment when you first pictured 20% of the neurons in your visual cortex quietly activating, restoring a pale copy of a long-lived evening. Maybe the smell of tangerines, which we started with, will now evoke a slightly more complex emotion in you, because a layer has been added to it tonight. A layer of knowing what exactly is happening inside your head at that moment. That the cascade from cortex to chromatin is running right now. That your brain is quietly rewriting itself. And that tomorrow morning, you will be a slightly different person than you were when you lay down. Not because anything changed around you, but because your memories, and therefore you yourself, were gently rewritten overnight. Claude Bernard, the great French physiologist, once said that the constancy of the internal environment is the condition for a free life.
Your body maintains temperature, pH, glucose levels, blood pressure, thousands of parameters within narrow limits so that you can live. But your memory does the exact opposite. It does not seek constancy. It continuously changes, adapts, updates, erases the old and writes the new. And in that continuous change, in that stream of rewrites and reconsolidations, is born what we call I. Not a fixed point, but a process. Not a photograph, but a river. You do not need to remember all of this. You do not need to hold on to a single one of these thoughts. They are already there, inside, being processed by the quiet machinery that evolution has been perfecting for 4 billion years. All you need to do is let it work. Close your eyes. Let your thoughts soften at the edges. Let your breathing slow. Let the hippocampus begin its nightly work of sorting and sending. Somewhere at a level you will never see or feel, your neurons are making decisions about your past. Which moments to keep, which to release, which to rewrite in light of new experience. Those decisions are being made for you, without your participation, by rules that were worked out long before creatures with brains appeared on Earth. You are a memory that has a brain, the most complex and most elegant expression of a 4 billion year project by life to preserve its past. And right now, that project quietly continues. Inside you, in the dark, while you rest, breathe, let go. Your brain will take care of the rest.